All right, guys. So we have a
Okay. Yeah, no break.
no break. Really?
Okay. Okay. So let's start the next session. And unfortunately, we don't have time to break.
Yeah. Everyone, remember, you can pause YouTube temporarily while the session is live, and then start it again if you have, if you'd like to when you get back. It does pause for a certain period of time. I am going to give this over for the introduction of the session by Diana, and can't wait to see that.
Okay, thank you, Joel. So I actually have the pleasure to introduce and moderate this panel. And so with no further ado, because we're running pretty late, so let's see if we catch up. I want to introduce Diana Bonilla. It's a great pleasure to introduce her. I've known her for several years, and I met her exactly at FlowTex. And we actually spoke in the same meeting in 2018, I believe. So Diana Bonilla Escobar is an immunologist with a PhD from Texas A&M University. She has a postdoctoral degree from Baylor College of Medicine and an ASCP accreditation as a cytometry specialist. She has more than 20 years of experience, although she doesn't look much more than 20 years old, as a biomedical scientist for a variety of applications, including infectious diseases and cancer at MD Anderson Cancer Center. She was one of the Isaac SRL emerging leaders and is highly involved in Isaac educational activities and task forces. So she currently works as an application specialist for Cytec Biosciences. And it is with great pleasure that we're going to hear her talk about high quality data in, high quality analytics out. Thank you, Diana.
Thank you, Rui, so much for the kind introduction. So happy to be here. I've been part of FlowTex for more than a decade, so happy to see that we were able to pull this one out virtually, and hopefully we can see each other in person next year. Just want to make sure that you guys are seeing the correct screen. That's
yes,
the case, Rui? Perfect. Yeah. There you go. Well, thank you. So for data analysis, the old saying also holds true: garbage in, garbage out, or a little bit nicer, high quality data in, high quality analytics out. And especially now that we have seen all the current innovation in reagents, in instrumentation, the measurement of more and more parameters simultaneously in the cells for millions of cells, multiple samples, so very complex data sets. So there is a clear need for robust data analysis tools that allows us to explore the data sets and also understand and capture their complexity. But the key point for the talk, at least my part, is that before you even think about the different computational analytical options that we will see later, you need to make sure that you have rock solid data that you can trust, that is reproducible, and that's again the main goal for today. I'll review 2 steps that you need to follow before jumping into any computational analysis. First, guidelines on how you can generate good data, and especially how to assess the quality of your data. And then a few background slides on computational cytometry before we hear more from Tim and Ryan. So the big question is how to make sure you have good data. So the first critical consideration or something to remember is that high quality data means that all your populations are easily resolved, especially cells with or populations with dim expression, rare subsets. So at least for fluorescence cytometry, you want to make sure the photons that are emitted by your fluorochromes, your fluorescent proteins, are efficiently captured and also resolved from noise from the background. So first is that you need to have a cytometer that allows you to resolve well your different populations. I'm going to walk you through multiple concepts using an example. This is a panel defined or designed to evaluate immune responses and to characterize multiple hematopoietic cells, multiple compartments, and within each compartment, multiple subsets defined based on the expression of different markers by those cells in the samples, marker that allows us to define memory status, activation, exhaustion, and once again, lineage markers. let's say in order to capture the complexity of all these different markers and subpopulations, we design a 40-color panel. So it is a panel that allows you to capture or characterize 40 different molecules in every single cell by using fluorochromes that have differences in excitation emission properties. So if you think about having only one marker in your panel, as Rui was mentioning, the analysis is simple. You can define two possible combinations, negative events, positive events. But when you increase the number of colors in the panel or the number of dimensions that are being evaluated, the complexity is clearly increasing to the point where you have 40 colors, 40 dimensions, trillions of combinations, multiple potential subsets that can be identified based on the negativity of positivity for those different markers. And it's not only the complexity based on the markers and the study per se, but it's the increase in all the resources that are needed to analyze the data. Time from the operator, all the expertise in terms of bioinformatics that is needed, the computational power that you will need, and an understanding of the different packages and softwares that are available. And just to give you an example, just to continue with that 40-color panel, if we want to try to gate manually that 40-color panel and just to identify the main subsets, you ended up with a lot of 2D plots. Yeah, sequential gating this is what we traditionally do using 2D plots. But this reveals the complexity of these big panels and the limitations that manual gating has. It's time consuming, it's error prone, it's very biased by the knowledge of the investigator. And in addition to that, it doesn't capture the complexity of the data because we usually, when we do manual gating, we tend to focus on gating only the subsets that we know. So you miss the potential of discovering new relevant populations for your particular study. So what do you need to do then? Then there's many computational tools that simplify that workflow and allow you to do kind of what manual gating does in a simplifier and faster way. What you would need to find, it's a tool that allows you to define what are different subsets that are present. There are multiple approaches to do that. Just to give you two examples, but again, there are so many other ways to do it. One of them, as Rui mentioned, clustering. Clustering is the process of grouping cells together. Based on what? Based on the expression of all those 40 markers that were characterized by, for example, this particular panel. And then you classify the cells based on the subsets and based on the markers into different clusters. The second most known way to evaluate the data and capture in one single shot the complexity of the sample. The other way to do it is, remember, we start with a 40-color panel, that's 40 dimensions. So try to reduce the dimensionality of the data sets to 2 dimensions and define a graph where you can plot those 2 new dimensions. So you basically create a new value for every event on the x-axis, on the y-axis, and then define the position of a single event in that 2D map based on their marker expression and how similar they are. So islands are going to be formed by cells that have similarity in the expression of those 40 color markers. And then the distance between the points will reflect that similarity. So multiple ways to then capture this and be able to extract the information from the samples in a more objective way, understanding still partially the complexity of the data set. And something important is that after you apply clustering or dimensionality reduction, what you're going to have is extra information about your events. So if you open an FCS file on a CSV format, let's say Excel, you will see that every event in your sample was characterized based on the expression of the different markers, 40 in this case. But in addition to that, when you run clustering or meta clustering, you will know for every event what the cluster it belongs to, again, based on all these different markers. And also for dimensionality reduction, you will have extra pieces of information. Let's say if you use Disney or UMAP, and we will see the details of all these algorithms with Tim and maybe a little bit with Ryan. But the point is that now for every event, you will have a coordinate on Y and X axis for when you do dimensionality reduction. But at the end is information, extra information you get from your events and your ability to plot that information and use it for any downstream analysis or comparisons between events, between groups, between samples, or between subsets within the same sample. So to reach that point where you have good data in order to use any of those computational tools available. You need to start from the beginning. First steps is definitely have a good panel design. You assign the fluorochromes based on their characteristics, their brightness, their uniqueness, their spread into each other, and that you and also based on the markers you want to quantify, based on the antigen density, based on co-expression, that you also validate your antibodies there and you titrate them well as validating every single reagent, buffers, and other things that are used during the staining protocol. Optimize your single color controls, the ones that will be used to train the software to understand what fluorochromes are present in the multicolor sample. Independently if you do compensation and mixing, it's important to go through the process of optimizing your single color controls. Optimization of every step in the staining protocol. Your buffers, your washes, your incubation times, fixations, define if you need sequential staining to improve your resolution for some of the markers. And the last part, it's make sure you put all that together, your panel, your controls, your staining protocol to have a panel that is performing well. And again, that's reflecting in how well your populations are resolved, even in the context of having 40 markers, 40 fluorochromes interacting or marking at the same time. So the point is that after you validated your assay, you want to make sure that every time you use the panel, the data you obtain is trustable. And we have these 6 steps to assess that. And again, ideally in every sample or at least in every batch that you're running. Make sure that the instrument works well. Your settings for acquisition are correct. Your controls are giving you good spillover calculation and are good quality, that you remove any unwanted events, and that you check, especially from the biological point, if your panel is performing well or not. So I'm going to go one by one. Step number 1, check the instrument performance. Run daily QC, review the report. Remember to allow your instrument to warm up fully, keep it clean, remove any residual detergent. QC passing alone is a great indication that your instrument is performing well and that you will have good data if you optimize all the other steps. Also check every parameter in your daily QC across time to make sure you have an instrument that is performing stably. Step number 2 is check your instrument settings, the ones that you use for acquisition. And that includes gains, voltages, thresholds, area, scaling factor. You want to make sure your population is on scale. You don't have out of scale signal. You want to make sure your signal is stable across time for every scatter parameter, and definitely for the fluorescent ones. And for every fluorescent detector, you want to make sure the gain value, the voltage value that you use gives you that perfect separation or resolution for the different colors in the panel. And not only that, but if you have a big data set, multiple samples run together, make sure there is consistency across the numbers for value and also signal type, in this case area, but applies to height and width for the electronic pulse. Step number 3 is your controls. These algorithms for compensation and mixing are fed by the single color controls. That's how we train them in understand what's FITC versus what's PE, and the same if you have 40 colors. So depending on what instrument you're using, you might need or not an unstain. If you do, make sure your unstain has a clear signature. And if you're using single color controls, general guidelines for every technology is that you have a positive that gives you the expected excitation emission profile, that positive you check that doesn't have any fluor mismatch, contamination, tandem degradation, also carryover, and that is bright enough to give you good calculation for your sample. Once you do that, you run your compensation, you want to make sure that it's accurate and it's not giving you false positives or extra negative events because there are errors in the calculation. How you would do that? Then you want to plot every fluorochrome versus all the other ones in the panel, all the permutations. And the goal is to identify or confirm that the negative population and the positive are perfectly aligned. If you see populations curving up, under-corrected, curving down, over-corrected, something that needs to be fixed by getting better controls. Step number 5 is to start removing unwanted events that are going to be misleading in your computational analysis if you don't define them on time. First thing is see how much the signal is changing over time. So if you plot time versus every single fluor, you should be seeing stability. If you don't, if you see spikes, if you see gaps, if you see fluctuations in the signal, there might be a problem with the instrument warm-up, the fluidics, even degradation of your reagents, but it's something that needs to be identified, excluded ideally, or corrected for. So make sure you take the time to review that as well. So doing this manually for one sample, it might be doable, but it's definitely not effective for large size or for small chips like this one, where you cannot simply gate out the good parts or the gating the good parts and exclude the bad ones. It's really difficult. So there are tools, computational tools that allows us to do that. I'm gonna briefly talk about two of them, FlowAI, FlowCat. So I'm sure Ryan will tell us more about his FlowClean. But just briefly, what FlowAI allows you to do is remove problematic events. Problematic events in terms of flow rate, changes in the flow rate, in terms of dynamic range. So you can exclude negative outliers. You can exclude also those events that are in the upper margins of the plots, very bright events that are also anomalies. And also you can evaluate, or the software or the algorithm will allow you to evaluate signal acquisition for every detector, every fluorochrome. And if there are areas where that signal is fluctuating, can be also excluded depending on your settings. For FlowCat, something similar. It's able to identify and remove areas where the density of the events is different, where there is also drift of the signal, and will remove at the end events that don't fall into certain range, and again, allow you to use only the good part of the events for downstream analysis. And it's not only related, when I talk about removing unwanted events, it's not only about instability in your fluidics. It's also cell debris, again, the marginal events, doublets, aggregates, also antibody aggregates, and removing dead cells. And why that is important. So if you remember my explanation about dimensionality reduction, in 2 dimensions, I want to see all the events in my sample. And then based on their similarity, they will be grouped together in islands. We are seeing the different islands identified there. These are all the events in the sample. If I can start showing you how much contribution all these anomalies or unwanted events are going to have into your real populations, all the doublets for forward scatter, for side scatter, adding events that are going to be misleading in your populations of interest, even more clusters or, islands identified just from the debris, or all these bridges identified just from having the dead cells present. So make sure you have a clean population to start with. And important also that you do a proper scaling of your data. So this is the same example, same sample, just displayed with different transformations or data scalings. For example, logarithmic, and the one that is going to be critical for today, the R sign transformation. For add sign, you depend on a cofactor. You can define that cofactor on your own. And these are numbers for that cofactor. And just to show you how different it's going to look depending on the number you use, 6,000 ideally what we use for spectral cytometry. Just to show you, if you go too low on the cofactor, what you are aiming for is to have symmetry around 0. Then you adjust the cofactor in terms of that. If you adjust it too low, you ended up splitting the negatives, for in this case, your CD8 population, into 4 subsets. And that's what the algorithm will see. Multiple populations identified there, not because of the biology or because of a real functional subset being identified, but because of the way you scale the data. And this is to reinforce that concept. Let's say we use a cofactor of 6,000 and we see the negatives in this plot for CD11C very tight. Lower, more separation, and even lower separation into 4 subsets, which is just the real negative for this marker. If I put that into an analysis tool, in this case using UMAP and using color-coded information, or to see the expression of CD4, I see how the CD4s are pretty tight, versus CD4 splitting in 2 islands, just given by the issue with scaling. And even worse, more separation, again, not given by the biology, by the findings and the populations in your panel, by just by the scaling. The final step is check your panel performance. Make sure that for every marker you can detect positive signal, ideally with a positive control if it's low on your samples, and that your signal is on scale and that it makes sense from the biology. Your 14 positivity out of the high scatter, your PD1 out of the low scatter, and the same for every marker. And of course, part of the validation of the panel initially. And also make sure that that makes sense. And as if I'm getting on CD3, CD4 positives, just to give you an example. And then I define memory subsets where naive and EM is what I'm going to be talking about, the vector memories. So if you have markers in the panel that allow you to confirm the nature of those populations and the identification correctly. For example, naive, you don't expect to see a lot of PD1 versus in the effector memory, even in the central memory, you start seeing that marker being more expressed. So kind of internal controls in your panel, in your sample that allow you to judge the quality of the staining and in general, the acquisition. So just a brief summary. And as a reminder, in blue of all the variables that are going to be important to control, to reduce variation in your data, and to have high quality data to then put it in any of the analytical solutions that are available out there. So if you have done that, a good panel, good testing, good optimization, and good evaluation of the data every time you run the panel, you have half of the work. But the other half is now knowing which analytical tool I should use, how I can use it, what algorithm, what pipeline, what software go for in terms of automated data analysis. And that's part of the goal today. And it's especially difficult because there has been a surge in the development of computational tools to overcome all the limitations of manual gating. So it's important to know what are the guidelines to decide. So if we go back to that 40-color panel, that complex manual gating, what we ended up having after using computational tools or for clustering, for dimensionality reduction, was be able to capture the complexity of those 40 markers and those multiple subsets, cell subsets identified in one single plot. In this case, we ran FlowSum and then UMAP to see the clusters identified by FlowSum or the meta clusters. And what you will see again is in one 2D plot, the multiple populations that were identified and then were characterized based on the markers in the panel. An important thing also, or two more important things, is that in the same map, you can evaluate other things, how different your donors or your subjects are, if there are any clusters that are more frequent in one versus the other, or changes in the intensity of expression. And in addition to that, the map will allow you to visualize the expression of the different markers using again color-coded images. We see, for example, all the CD4 red indicates high expression being in this part of the plot, and that corresponds to all the CD4 subsets that were identified, once again, in detail based on all the markers for activation and memory that were used. So that's the goal. How do you get there? Everything starts independently of the technology with an FCS file that contains every event in your sample characterized across multiple detectors and fluorochromes and markers. You first need to start with data cleaning, as I mentioned before. You then identify a method to characterize the populations, the different subsets in the sample, and ideally to be able to correlate that with the metadata of your clinical study, of your experiment, any characteristics about the study, responders, non-responders, and treated, and stimulated, different subjects, different time points. And ideally, after that, we all hope that we can identify biomarkers or help guide clinical decisions or treatment decisions with any of the findings. But independently of what step you're thinking of, you can do it in an automated way. There are tools, there are software, there are algorithms that allow you to do it. And all of them use the same basic unit, the algorithms. And algorithms, just to define it, is a sequence of instructions to solve the problem. And in cytometry are really useful because they help us analyze highly complex dimensional data, usually coded on R, Python, MATLAB, but they are also available through commercial software. And there are so many algorithms that can be used for every step in that workflow that I defined. We have cleaning algorithms, I like the ones I described, auto-gating for normalization, for clustering, so many more. Same thing for dimensionality reduction. There are so many others for trajectory mapping, for population labeling, and many other things, and others to do the statistical analysis and predictive and try to establish correlations, compare between experimental groups, and get information out of what you did with your clusters, the clusters that were identified, their frequency, and their expression of the different markers. The good point is that you don't need to do it on your own. There's so many options. Well, if you're good at programming, you could go for R, MATLAB or Python and find the scripts, bioconductor, create your own. And traditional software that also have these tools for clustering and dimensionality reduction. And we will see more from Tim. Some options that are cloud-based and also incorporate multiple algorithms there. More sophisticated ones that are personalized based on your panel, based on your analysis, that can even do the analysis for you, and some other ones that have a database of samples that have been pre-characterized and you can compare versus your own sample to guide your decisions in terms of what populations are being identified, especially towards diagnosis and treatment. But I guess the key point is, how do I decide which algorithm should I use? What platform should I use? So it's going to depend, and it's always the answer, on your data and your scientific question. But in general, you want to consider multiple things. There is something reproducible. If you run the algorithm again, if you run that data set on another algorithm, you find the same population of interest or the key finding in your conclusions. Processing time. You need something that has low runtime or computational efficiency. Important also your computational skills. If you have technical expertise, you remember if you want to use R, you require programming abilities to adapt these scripts or you to create your own. So that's going to be a limitation. But there's so many software in applications and pipelines with or even packages with their own graphical interfaces that you can just start using without having programming abilities. And a lot of things to consider from your data set, and I'll be quick because I know I'm running out of time. How many samples you want to process? Is this the one that you decided to use too slow to process your 500 samples? How you need to plan, and ideally having a biostatistician and a bioinformatician involved in the process of experimental design. How many subsets you want to identify? What's the frequency of your population of interest? If you have something you're tracking or instead of fishing, if you want to define the number of clusters or subsets you want to get at the end, an automated way or manual, supervised versus unsupervised analysis. If you care about the local or the global structure of the data, if you're thinking, because it's a large study, to add more files to the analysis, how you can do it, or if you can do it. And definitely a lot of considerations if you are talking about longitudinal studies, how to make sure the tools are going to work, especially if you have batch effects, and whether you need normalization and how to work on that. And I'm not going to give you details about the algorithms because a team will work on that part. But at least for now, I want to give you, and this is my last slide, a take-home message. And it's good practices, independently of what option you decide to use from now, a commercial software, programming, or even going for your own strategy. The point is, make sure you explore the data in multiple ways to define the best approach, at least initially. So you make sure you find the correct tool for your data set and for your scientific question. Make sure it's not only applying or using an algorithm, it's making sure the settings that define the results are going to be optimized as well, all the hyperparameters. That you evaluate the population you're finding as critical for your conclusions. It's stable. Be careful about fragmentation. You can go over clustering just to make sure you identify any population and then find clusters of clusters, or then changing the settings for your evaluation to polish your data analysis. And important, if you get a population that is critically defining responders versus non-responders, make sure you can find it through manual gating if you go back to that FCS file. And this is just to show you how same data set can look differently, at least for this too, when you change the parameters, and how important is the algorithm optimization. And I think that that's the end of my presentation. Thank you so much for letting me be here. It's always a pleasure and an honor for me to participate. Let me know if you guys have any questions.
Right. Thank you, Diana. That was a wonderful talk. Extremely useful.
Thank you.
And actually, there are a few comments there on YouTube saying exactly the same. So unfortunately, Diana will be able to stay with us for the roundtable discussion.
I might be able to stay.
You may be able? Okay,
yeah,
that's great. So then we'll keep the questions, because there were some questions that were coming up.
Good.
Those questions for the roundtable. And so we'll move forward then to the next speaker. Thank you, Diana.
Thank you.
So our next speaker, is he coming up? Yes, he is. So our next speaker is Timothy Crawford from FlowJo, BD Biosciences. So I've known Timothy also, or Tim, for some time. We've met, and if you guys have been attending these flow cytometry meetings, you know that Tim is usually in these meetings, always very useful in providing a lot of good information on data analysis. So let me just say a very brief words about Tim. Tim received his PhD in molecular and cellular biology from the University of Washington in Seattle, studying developmental genetics, stem cell biology, and cell signaling mechanisms. He gained additional training and experience in HIV biology, viral immunology, and cytometric data analysis as a member of the UCSF core immunology labs in San Francisco, then joined the Hawaii Center for AIDS. That must have been nice, if it is in Hawaii, as a staff scientist, where he worked to evaluate the phenotype and function of T cells, NK cells, and monocytes in HIV-infected persons. Currently as an application scientist with BD, Life Sciences Informatics. And yeah, Tim provides a lot of training and assistance to scientists across the globe on single-cell data analysis approaches and techniques. So Tim, the floor is yours, where you're going to be talking about algorithms for dimensionality reduction and clustering.
Thank you for the introduction, Rui. I appreciate it. And Diana, excellent introduction here to algorithmic processes and controls that are needed, and the steps needed to get good data out of high parameter, or good analysis out of high parameter data sets. So this is a non-commercial talk, so I'll just say I'm, a senior application scientist with the BD Life Sciences Informatics Group, formerly known as FlowJo, and I'll just be giving you my opinions and insight onto how to properly utilize some of these clustering algorithms and dimensionality reduction techniques that are available to mine high parameter data in a meaningful way. And so as I see it, the basic goals for high parameter analysis are to define your populations within your high parameter data space, visualize the phenotypes of those populations, and then enumerate the differences in populations between your experimental conditions. And of course, you can do that with standard traditional hierarchical gating to define the populations that you know. But you can also run a computational algorithm to subset your populations and classify them into distinct groupings, which are known as clusters. And each of those clusters has a distinct phenotypic expression pattern that you can visualize in standard plots, either univariate or bivariate plots, one at a time, overlays like I've done here. Or you can enumerate all of the median statistics as measurements of central tendency for each channel, for each population, and get an idea of what the expression pattern is. Ultimately, you do want to understand how those populations change between different conditions. If you're stimulating the cells, do these markers go up or down? Do you lose populations because they gain expression of other effector molecules? They change their phenotype. And you do need to be sure to manually gate your populations. I think manually gating is an essential step because you know the phenotypes that you want to focus on. You have a hypothesis, you have a scientific goal in mind, and you should gate for what you know and answer that fundamental question the way we already know how to do, which is hierarchical gating. But the problem with high parameter, as you get into 20 plus colors, is that you end up with all of these additional markers downstream of your major subsets. And maybe you're looking at a slew of cytokine expression profiles like this OMIP panel from the Rotor group. Or maybe you're looking at exhaustion markers, but either way, what do you do with this data? Do you evaluate the frequency for each memory population, one marker at a time? Do you do combinatorial Boolean gating for 8 markers downstream of each of these memory populations? That's going to give you 256 combinatorial plus minus gates to wade through. So you're now working with thousands of potential phenotypes, which again require some sort of high volume processing and algorithmic approach to actually pull out those meaningful phenotypes. And is that even going to represent the true biological state of your system? You only gate for what you know, so gating is subjective, and you cannot go through every single bivariate option and actually find populations in a manual way. You're never going to know everything about your system. You may be using new markers. And so this is where those unsupervised clustering approaches have come into use in the field over the past 12 years. There's over 30 different methods that have been developed and employed with varying success on biological data. Some of these methods are trained or designed for flow cytometry data, others for mass cytometry data. Some can be used on both data types. And they do different approaches mathematically to classify your data. Some are peak finding algorithms like DenseVM and Accents. And in that case, you actually have to do the dimensionality reduction first, give you a dimensionally reduced data space within which to peak find and classify dense regions. Others actually give you differential expression endpoints that say, oh, this cluster is actually correlated with my endpoints, which can be useful. And then yet others have visual representations like flow, sum, and spade have become popular because they give you minimal spanning trees and meaningful information in the form of the visual aid to interpret the clusters that come out. And you have to start somewhere. So I recommend anybody that's new to the field to go ahead and pull up a review, something that's fairly new, and go through the history of these clustering algorithms. In this case, I'm just showing from this review a figure where they classify the number of citations in high-end journals using these different algorithmic approaches. And you can see some are rising to the top as being very useful, while others have kind of been around for a while and have kind of gone to the periphery and people aren't using them as much. Some of that has to do with their accessibility. All of these tools are available as packages within R or Python. But if you're limited to a commercial software, you're only going to have a subset of those tools available to you, which is more than enough to mine data and explore it. But you will be limited to the graphical user interface of the program. So all of these algorithms really do the same essential thing, which is subset your entire system, all your data points, into populations in a kind of unbiased and objective way. And I think this gives you a good summary of the distinct combinatorial states within your system without ever manually gating down to a specific population. You can go ahead and run an algorithm and you get a whole bunch of complex populations out of it. And then your job is to find those endpoints that are meaningful within your biological system, within the question and hypothesis that you're entering this study with. As I mentioned, you can enumerate the median values of all of the markers for each population. You can also look at the traces and call phenotypes one at a time. It's kind of hard to see all the phenotypes when you overlay them all together when you've got 30 plus clusters coming out of one of these algorithms. But some resolve populations quite well. Gamma deltas and FOXP3 Tregs pop out as single populations here. But other expression profiles are going to be continuous and variable depending on the population. We see numerous clusters with intermediate or low levels of expression with CD95, CCR7, CD27. And my point here is that a clustering algorithm has the ability to pull out intermediate transitional states, grouping those cells together based on a median fluorescence intensity that's different and stair-stepping through that smear. If I were to just draw a manual gate right here through my positive and negative, calling everything on the right positive, everything on the left negative, I would be cutting through those low dimensional or those low expressing clusters and bifurcating them and maybe completely missing them with a manual gating approach. And I think that that's going to, what's in the chat here? Oh, sorry, okay, just double checking. And I think that's going to, really not give you as much depth as a clustering algorithm will. So remember that clustering algorithms are just to subset into populations in a kind of unbiased way that's driven by the math, and it's reproducible in that it's the same math every single time, and it's not limited by your knowledge or your manual gating approaches. So where do the dimensionality reduction algorithms come in? Well, dimensionality reduction is potentially just a way to visualize your clusters. You can overlay them into this dimensionally reduced data space and show them to your audience in a meaningful way that shows all the data in 2 dimensions as opposed to 25 or 40. But there are also additional uses for these dimensionality reduction algorithms independently of clustering, because dimensionality reduction really just gives you a low dimensional embedding, they call it, a 2-dimensional representation of your data, redistributing events into this 2-dimensional space. And the goal here for all dimensionality reduction algorithms is to present your data in fewer dimensions while preserving the high dimensional structure of the data as much as possible. And you can see that by taking your manually gated populations and just overlaying them onto this dimensionally reduced data space, and events that are similar to each other should be grouping together within this space. You'll see that with overlays of your lineages. You can also turn on a third parameter, heat map of third parameter across that data space to orient you and focus in on the various areas or regions where you see expression of your various markers of interest. But you don't have to always look at everything. You don't have to zoom out and see your entire system, all the lineages together. It may be prudent to actually gate away some of the cells, cleaning up your data down to a specific lineage. And this is, again, always dependent on your experimental question and what you're focusing on with the panel that you're employing here. In this example, 25 colors, whole blood, if you use just all the cells, I mean, this visualization, this T/E downstream of just CD45 positive gating is dominated by the granulocytes. And all the other lineages are pushed off to the edge, and you kind of lose resolution to see the peaks and distinct populations within those lineages. So why not remove those grands and spread everything out so you can see the other lineages more effectively? And you can do that with effectively dump gating lineages using lineage markers, or drop all of those into the same color and get rid of all of the lineages at once, focusing in on the particular population of interest. Here I'm gating for CD8 cells, which are overlaid in blue, heat mapped up above, and you can see that, you know, it's spreading these events out so that I see distinct peaks or regions of density that I can then cluster on top of, or even manually gate apart and see what those features are. So don't be afraid to gate down to specific subsets and employ dimensionality reduction and clustering algorithms at any level in your gating hierarchy to focus in on a particular subset for a particular question. I'm showing you all these T/Es because this is what I was familiar with. It's the algorithm that's been out the longest and commercially available through different softwares. And so we've gotten used to seeing T/E plots, but it's not the only dimensionality reduction algorithm out there. Diana was showing a bunch of UMaps, and UMap is kind of the new kid on the block. The past couple of years has really come up in use in both cytometry and single cell RNA sequencing data. It's useful because it actually processes faster than T/E to some degree. It scales better with more dimensions. So when you go up to 25 or 30 dimensions, or even in a single cell sequencing experiment, hundreds or thousands of dimensions, UMap will process much faster than T/E. But they do have different, purposes here. UMap does a good job of keeping cells together that are similar to each other, that is preserving more of the global structure. This is due to the way UMap is initialized, the way the embedding is processed, the math behind it. And T/E has this problem when you start getting up into higher numbers of dimensions. This is only an 18-color experiment that I'm showing here, and it's already starting to occur where one lineage island here, I call this a continent-like structure, and each of these different regions is obviously grouping the events together based on their similarity of lineage. But notice how the B cell population is split here, and now you've got this population island off at the top that is completely separate from the other B cells. And as you go up in the number of colors, this will happen more and more to where you get all these different islands and they're displayed all over the place in your T/E. And so you can't actually infer the positional relationships, don't tell you anything about the similarity of those populations within a T/E space. Within an actual island, you can pretty much say those events are going to be very similar to each other because T/E does a good job of preserving local structure. And if this does split up, there's meaningful information in seeing these islands split into these these lineages split into various islands, it means that they're probably different from each other with respect to some other marker. And if you start mining your data, you're going to find things that are different. So this pops out because it's distinctly CD95 positive. But UMAP does a better job of keeping those islands together, but it doesn't flatten the data as much. T-SNE, you won't find populations that are kind of overlaid onto each other of different phenotypes. UMAP, you may find that there's a z-axis there and you've got multiple populations kind of grouped together. So that's one thing to consider. The other thing is how these algorithms process, even though they give you very different representations of your data and preserve local versus global structure differentially, they do process both in an iterative sort of fashion. So T-SNE takes high dimensional data, converts it into a Gaussian probability distribution, and then it seeds or initializes the low dimensional data space. So each event gets a position. It converts that low dimensional representation into the same Gaussian probability distribution and compares the two using relative entropy or the Kullback-Leibler divergence. And then each cell will find its nearest neighbors. They'll push and pull against each other, and cells that are similar will pull closer together, dissimilar push away from each other. And then again, convert to a probability distribution and compare the low dimensional probability to the high dimensional probability. And that's one iteration. And it will do that 1,000 times. T-SNE will find nearest neighbors, push and pull, convert and compare. Find nearest neighbors, push and pull, convert and compare. Until finally you get this resolved low dimensional embedding representation, this visualization that has all the events in 2 dimensions that you can mine and use as a visual aid to confer a message to your audience. UMAP uses different math. It converts into these fuzzy simplical complexes, creates a manifold across the data, embeds and initializes the low dimensional space, and then converts that to a fuzzy simplical complexes and compares these 2 representations of the data using a method called cross entropy. But it also does this iterative type of processing where it does this a bunch of times, finding nearest neighbors, pushing, pulling, and eventually resolving. Now, whichever one you use, you have to remember that T-SNE and UMAP need run on all the data together in your system in order for that representation to be comparable across different experimental conditions or different samples. And to do that, we actually concatenate the samples together, merge them all together so that you have multiple samples in the same single file. You can downsample at this point in order to normalize the number of events coming from different conditions, study arms or samples. And you can also annotate your samples with relevant metadata labels so that you can pull them apart by your different experimental conditions. And so here I'm concatenating, creating parameters for tissue and time point, and organizing my 16 different samples from 4 different tissues and 4 different time points, all into a single file and running T-SNE on that same file. At that point, you can then look at one of those condition parameters, gate those conditions apart, and visualize each of the separate sets of events from different tissues in this case within that common dimensionally reduced data space. You cannot think of T-SNE and UMAP or other dimensionality reduction algorithms as a gating approach. You can't run them on one sample and then apply that to different samples. You have to have all the data there together. You run the algorithm once, and then you pull apart the samples for visualization and downstream exploration. But you can do a lot with this visualization once you have it. Even without subsetting into clusters, you can create a gate. And this is why I like T-SNE, because it's a very flat, separated visualization. And so the different regions will be distinct populations. UMAP has a tendency to cluster or layer populations on top of each other that are within a lineage, even though they group together. So T-SNE, I like to make a gate, make an overlay. The magenta gives me the phenotypic properties of that gated region. And then, of course, I can take those gated populations across any of my experimental condition parameters and drop those gates downstream of this query gated population. And this will give me the distribution of those events, where they came from, how are they distributed across my experiment. Knowing what I put into the system using down sampling so that all of the cells are, all of the conditions are contributing the same number of events, gives me an easy starting point to interpret how those populations are changing across my conditions. I can also do the other way, which is gate apart the conditions and then drop the query gate downstream of each of those different condition gates. And then this will give me a gate within this space. And obviously visually we can see that the spleen has more of these CD95 positive B cells than anywhere else. So both are valid, but I like to think of it this way, where I'm trying to classify these gated populations or cluster populations, if we use an algorithm, and determine how they change or how they're contributed from the different experimental conditions in the concatenated sample file that we're exploring. Now, clustering will do something like that. It just subsets the entire data space without you manually drawing gates around peaks. And the clustering algorithms will produce a whole bunch of populations, and then you need to visualize them. You need to call the phenotypes, you need to determine how those clusters change. And so my method was always to export the cluster populations that come out of an algorithm as individual FCS files, drop them into a group in my workspace, add the MFI statistics, so all the medians, count statistic to know how big the population was, and experimental condition gates to know how they change across my conditions, and then enumerate those stats in across a group with a table. Of course, a little formatting in Excel, and now I have something meaningful to share with my group at lab meeting, and that is the actual population phenotype, what it's expressing, and how it's changing across those conditions. And that can be done, by anybody that has relatively little experience gating and applying statistics and enumerating those in a standard commercial package. But everybody's always asking, well, which algorithm do you use, right? And I can't really give you a solid answer there. I can say that the community has started to be drawn to certain algorithms for different reasons. FlowSum has been a favorite of the community because it analyzes using the self-organized map and k-nearest neighbors to overcluster, creating by default 100 mini clusters, and then the user specifies the number of meta clusters to come out. So I can say I want 32 populations, and I'll get these different colored populations that are groupings of the mini clusters together that represent the actual population downstream that's overlaid here in blue. In this case, in terms of the B cells. FlowSum is super fast, so you can classify millions of events if necessary. And it produces some nice visuals. These minimal spanning trees with radial star plots can be super helpful. You can apply the map that you create to other data, so you can iterate across your conditions and get different maps that show relative expression levels. And it does push out, the medians and a heat map of the dendrogram to show you the relationships of those clusters. Not all the features are available in every implementation in commercial packages, so you can't do everything you can do in R, but this has been a really useful tool, and we can see that in the number of publications and how many people are interested in employing it because they're starting to see it be used in their favorite journals. The other algorithm that I am really excited about is Phenograph. And Phenograph has gained some notoriety in how it's used in single-cell multi-omics or single-cell RNA sequencing data. It constructs a nearest neighbor graph and then creates communities using Leuvian community detection. And it overclusters, has a tendency to give you more populations and subsets within lineages than FlowSum or some of the other methods. One of the good things is that you don't need to know the complexity. You don't have to say how many clusters does it come out. It automatically determines the number of clusters. And while it's a lot slower than FlowSum, it is fast enough to scale up to hundreds of thousands or even millions of events without downsampling, though you will need to wait for it to complete with a significantly increased time compared to FlowSum. There aren't any default visualizations that come out of Phenograph, so you're going to be on your own to represent the phenotypes in some meaningful way to your audience. But I do think that it's a really useful tool that people should be looking into and considering when they want to use a clustering algorithm that is an unsupervised approach. Different methods and actually compare tables showing the Phenograph. Traditionally, we use F. You can all Aryan algorithm, and a lot don't actually use hard numbers, but there are a couple that use F measure statistics. And what you'll find, they have different math underlying. Maybe 60% of the clusters will be matched well, and then the rest of them kind of trail off. They also won't match your manually gated populations because these are unsupervised approaches. You aren't telling this algorithm that Z3 is meaningful and what you expect downstream, the way your brain does as an immunologist, knowing those populations. So I wouldn't expect to recapitulate manual gating here, but I would just try and think about this as a data mining. You choose rhythm, you use it to create these populations, and you get a summary of all of the different combinatorial states that you can then mine for more information depending on your scientific question and your goals and your aims. This is really why these methods have become employed in multi-omics data and high dimensional data that has hundreds or thousands of parameters. Then you can't even gate for what you know. You have to go right into dimensionality reduction to visualize all the populations, and clustering to subset into novel new things and discover what's going on in your system. And so when you have hundreds of parameters, you can't go right to T/SNE. You have to go through principal component analysis, go from hundreds of parameters to a limited number of principal components, and then go from those principal components into T/SNE. UMAP has been really popular with the high parameter guys in single cell sequencing because you can just take all the features and you can go directly to UMAP to get this overview of your data structure and compare what's going on. But this may not give you the resolution you need, so don't be afraid to zoom in on subsets using alternative methods. Here, I focus in on the NK cells and use another method called TriMap, which is even the newest option that I have available to me. And it does a good job of showing developmental trajectories and connections between populations. So I can spread out that subset using a different approach, cluster across these events to give me unique combinatorial expression states, do differential expression analysis to find those combinatorial or those genes and proteins that are up and down regulated. And ultimately, I'm looking for a signature, a combinatorial expression pattern that is meaningful within the biology of the question I'm asking. And I think these are very good tools to mine data in a kind of an unbiased way to see what's there, but don't expect it to be reproducible. If you run it over and over and over again, there's going to be slight variations in most of the methods and what you see. But it certainly is a valid approach to look at your high parameter data, and it should be a complementary approach to manual gating, not a complete option, not a complete replacement of that. So in summary, T/SNE, UMAP, these are great tools to visualize high parameter data in lower dimensional data representations. Both of these tools produce biologically meaningful embeddings that can be useful, but T/SNE generally preserves more local data structure and UMAP preserves more global structure. These unsupervised clustering algorithms, whichever one you use, they can be helpful to identify distinct combinatorial expression states. And I think that's the most useful, is you're just mining that combinatorial expression to see what your system is providing you. But these algorithms do still present significant challenges, not the least of which is the accessibility of all the algorithms. Not everybody is going to have computational backgrounds and be able to program in R and use all these tools. All of the tools generally come out with clusters, but they don't tell you what those clusters are. So there's a need to manually annotate for the biology. You're going to have to go and identify the expression pattern and say that these are T effector memory cells, that these are activated, these are exhausted. Still based on your own biological expertise. There's a distinct lack of interactive visualization tools, so you can click on a cluster and explore it more. You know, some of these clusters just come out as stat populations, and depending on the package you're using, you can have more or less interaction with those visualizations. And of course, there's always a need for thorough comparisons of these evolving methods, because even, you know, in the past couple of years, we have new dimensionality reduction techniques that preserve the relationships of the data differentially. And I think it's an opportunity for maybe new people that are coming into the field and learning about all the options. As they do explore the options, they can do comparison publications and actually contribute to the community's understanding of which ones are the best to use for certain types of data and certain applications. And so with that, I'll just say thank you to my bioinformatics team and all the data authors and algorithm authors, and of course, all to the countless others, including the FlowTex community, for inviting me today. Thank you for your time and attention.
Tim, brilliant. Thank you so much. I mean, it just shows, you know, that we're still in such early stages of really understanding all these tools. So having, you know, people like you and the rest of the panel, you know, giving these kind of presentations is just so helpful. And I've seen a few questions coming up exactly on this. So yeah, so let's wait for the panel discussion and we can try and answer some of those questions. So let's move on then to the next speaker. And our next speaker is Ryan Brinkman. I think for most of you that are involved in flow cytometry, Ryan doesn't need any introduction, so I won't give any, no, I'm joking. I will give an introduction. I have something written here. And, you know, it's a great pleasure to introduce Ryan. He's actually been a speaker at FlowTex a few times. And so Ryan is at the British Columbia Cancer Research Center. His research is focused on developing and applying flow cytometry bioinformatic approaches to advance our understanding of human health and disease. His early work centered on creating the required data standards as chair of ISAC's data standards task force, and a very important task force, and a free open source computational infrastructure in R, now widely used to support high throughput computational statistical analysis of flow data. His recent efforts have concentrated around developing complete analysis pipelines to cover all the steps from flow cytometry data pre-processing to automated gating and biomarker discovery. And this is very exciting. And he's also been applying these methodologies as part of collaborative projects, primarily in the area of immuno-oncology clinical trials. And Ryan is also active in the community and leadership of Flow Repository and FlowCap efforts. He was also the 2018 ISAC Distinguished Service Awardee. So Ryan, it's a great pleasure to have you as our last speaker, where you're going to be talking about data-driven cytometry. So
thank you. I hope you can see my screen okay.
Yes, we can.
Excellent. Thank you for that fantastic introduction. Real pleasure to be here in my office at FlowTex. I appreciate the invitation. A disclosure, I'm involved with Cytopex Bioinformatics, Inc. Everything I'm going to talk about today is freely available. All the technology underlying everything I talked about, you can go get and use for yourself. So it's, I'm going to talk a little bit about something a little bit different than we've heard before, complementing what you've heard from other authors, other speakers today about unsupervised methods and dimensionality reduction. The problem I've been interested in is manual analysis. And why is this a problem? Well, everybody in this audience knows it takes a lot of time. It can be as much as an hour and a half. I've heard from some of our clinical partners in pharma. Even if it takes 50 minutes a file for some of these clinical trials, thousands and thousands of samples subject over time. It's highly subjective. There's a lot of variation between two people. There's a couple papers out there that have done some really in-depth analysis out there, but the numbers in the literature, 20 to 30% variation between experts in the field and the results. And that's kind of sad. And as Tim just said, you just can't analyze everything. There's just not enough time to look through huge data sets. And so that's a problem that we have with manual analysis. And so the whole, what I'm going to talk about today is the state of the art and how we're addressing the challenges for manual flow cytometry data analysis, where your goal is to identify cell populations to report on cell counts, percentages. Quite different than what we heard about for the goals we have with dimensionality reduction. Here you want to get, 25% of your cells are NK cells. And these are important questions, especially in the clinical context. So there's been lots of tools that you've heard about that are trying to address the issue of cell population identification. A lot of algorithms are unsupervised. And there's a nice review by Robert Robinson on 2016 that highlighted the performance of these algorithms. And we use something called the F measure, which is the harmonized mean of sensitivity and specificity that gives you a score of how well these algorithms are performing between 0 and 1, where 1 is you put every dot into the same box that's been done by manual gating. So we have manual gating as the absolute gold standard. A challenge with this is sometimes if your algorithm could potentially perform better, you can't actually show that by the F1 because that's going to be considered a difference, and we set the manual analysis as a gold standard. However, As Tim said, there's lots of algorithms out there. FlowSOM is still the go-to algorithm because its performance is really good on average. The average percentage you can see here is about 76, 77%. This is still not good enough for clinical use because sometimes the performance is quite bad, and sometimes if you're trying to make a diagnosis, if you're trying to decide if you want to put cell therapy into a patient, you're really going to be sure. And we know that manual analysis is good, so people haven't really adopted these unsupervised methods because it's just not fit for purpose for identification of cell populations in a clinical context, or when you want to have reportables. Great for doing unsupervised analysis if you're trying to do dimensionality injection, trying to find biomarkers, trying to get patterns. Not really good when you're trying to get cell counts and percentages. Machine learning algorithms have been developed, including from my group. We threw out FlowLearn a couple of years ago now. It's my stuff. I'm happy to say it sucks. There's other methods out there that didn't perform as well as our stuff when we did the comparison. And there's some good reasons for that that I'll get to at the end of my talk. There's hope. Something that does work is an algorithm called flow density that came out of my lab. This is a supervised method. And so the performance here you can see across a large number of different populations. Again, we're looking at the F1 score, including very small ones on the bottom right, is as good as it's ever going to get. Where we're comparing to manual analysis and some of the places you see in this graph where the performance isn't at 99%. That's because there's variation and subjectivity in the manual analysis that is impossible to meet. When we set manual analysis the gold standard, and when we look at those files where we're not having as good performance, it's because the humans are all over the place, and we can gate with flow density in a robust and reproducible manner, and on challenging populations for humans, we can gate those, I would say, better, and the score doesn't reflect the reality on those. But as you can see, by and large, performance is really good. The way this algorithm works is when you use this algorithm, has to specify the rules for how you want to gate your population. You set those rules once for your panel, and then you take your hands off, and the algorithm works. This is different than unsupervised algorithm where if you're using machine learning, the algorithm is trying to learn what you're trying to do, or if you're using a unsupervised algorithm where you have to tune parameters that you have to guess what those parameters are, and then the algorithm is trying to fit models to the data. And one of the reasons why these unsupervised algorithms are never going to, I think, get the performance that we can get with the supervised method is because in an unsupervised method, you have to make assumptions about the size, shape, and distribution of all the different cell populations you're going to see in a data set. And those populations look really different, right? You get smears, you get heart shapes, you get tails, right? Lots of different things going on. And with an unsupervised algorithm, you kind of have to fit a model to everything. So it's never going to be perfect. Well, with supervised analysis, we can really make things perfect, and we do. And then once you set that up, as you can see here, here's 5 different samples from the same study. These gates are going to shift based on the data that's presented to it, based on the rules you set. And so here you have to know what the heterogeneity in your data is, so you can define the rules. So in this data set, we know that there's either perhaps one population or 2 populations, and then we set rules that, for example, if there's 2 populations, we'll draw the threshold in the middle between those 2 peaks. And if there's only one population, we'll set it at the second derivative, here at this inflection point. We have a toolbox of different rules that we can use. There has never been a population that we have not been able to gate. I don't think there ever will not be. And it works fast. We can gate each file in about 60 seconds. And then we look at the performance of manual analysis shown here versus flow density on the other side. We did a paper in collaboration in 2019 over 11 different cell populations, 33 time points, ended up having about 1,000 different pairs. It's pretty much a straight line between manual and flow density. And the take-home message from the paper is that this flow density algorithm consistently and successfully reduces manual gating. And this happens again and again and again. Like I said, there's nothing that we haven't been able to gate. We have a perfect track record so far. This is one of the hardest examples that we've had. These are patients who failed first, second-line therapies. They're dying. There's all kinds of things going on in their blood. Really interesting story what they're trying to do in this study. They're trying to rescue them by throwing all kinds of drugs at these people to find something that works. But there's just a really messy data, and we were able to gate this so well that we're able to do better than the manual analysis was doing. And before we actually handed them the code, they started trying to gate like we were doing in the code because we're getting such good results. Because we enforce by having these rules to be reproducible and to be exact, and it just gets better data out at the end. Here's another example that I didn't think we were able to pull it off, but if you spend the time to parameterize these algorithms, this is one population here in this tetramer data. It only shows up in certain cases, and we had some test data that we knew what those were. But the algorithm doesn't know if those populations are going to be there or not. We have to set this algorithm up to say, well, if there's this population here, gate it. And then a new data set comes up and has to look, well, this is what one set of data looks like where there's not that population. There's no information that tells the algorithm that it's there or not, but we never gated it when it wasn't there, when we had the gold standard, when we knew that it was there or not. And then when it's there, we can pull out this. And this was a really hard population to gate. I don't think an unsupervised algorithm could ever do that. I could be wrong. I have perhaps reinforcement bias because we've seen this being used. 6 of the top 10 pharma are using this for ongoing clinical trials. It's being used to make diagnoses on people who have cancer. It's being used to test cell therapy products that are going to be put into people. So we've never seen it fail, and it's being used for clinical use. So we're really happy about that. So that's the state of the art where it is today. We have a tool that works. To address the downcomings, we have a manual gating where this, it's time consuming and subjective, the unsupervised doesn't work, but there's some problems with the supervised gating, not with the algorithm itself. It's fantastic and it's going to work. It's going to gate your stuff if you spend the time to set it up, but that's the key. You have to spend the time to parameterize that algorithm once per panel. And once it's done, your hands off, you never have to touch that again, provided when you set that up, you have captured the heterogeneity in the data set that you want to analyze. So you need to see your sick people, your health people. So you have some idea of the breadth of heterogeneity, but we know pretty much what population is going to look like by and large. And like I said, it's always worked, but it has to be customized. And the customization step takes time, right? So this is unlike an unsupervised algorithm. We just throw it at your data without pushing any buttons. So it can take, a couple of weeks or more to set this up per panel. I think our average is around 2.5, maybe 3 weeks per panel. It doesn't really matter how many populations you want to gate. You want to, gate a 40 color panel, it's going to work. It's not going to be any slower. Because we're just going to go through this. The reason why it's so quick and why it works is we go through this bivariate by bivariate by bivariate by bivariate, matching what you're doing by hand to gate those, but the algorithm is doing this instead. There's hope that within this machine learning, there's a couple kicks at that can, but it hasn't really worked. And I think that's because there's just not enough good training data out there. In order for machine learning algorithms to work, they have to have lots and lots of data. And that doesn't exist anywhere. We've been doing gating forever. But if you try and go and find a wealth of gated data, that doesn't exist. Even in repositories, it's really hard to get access to how people gated. And then remaining problems is 2021 is the new 2020. And we have this problem of COVID-19 happening around. So how are we going to solve these problems? Well, this is what we're working on right now. And I'm so excited to tell you about this project. It's the coolest thing I've ever been involved with, ever. Project Discovery is a collaboration we have with a lot of people, but mostly it's coming through with a collaboration we have with EVE Online. EVE Online is a massively multiplayer online game where we have thousands of, wait for it, it's going to make sense in a second. you have people flying around in space shooting each other. There's thousands of people online playing this game all the time. And interestingly, this is tends to be a more advanced gamers than you might otherwise expect. The average age of the users in this game are about 30 years old. Also, these are usually not 5-year-old kids. There's a lot of really macroeconomics involved in this game. There's a lot, you have to be really, there's a lot of intelligence involved in this game, just to say, besides just flying around in space shooting other ships. And the developers of this game, CCP, has also been really interested in science and trying to leverage all these gamers that they have to do other things and just fly around in space. And so this is idea of citizen science. And so what I'm going to talk about now has been done before by CCP and EVE Online, where you're doing a massive scale citizen science to improve some problem. And so there's been good success before. They got a paper in Nature Biotechnology, so it's good. And so when COVID came along, CCP was interested in what can we do to help this situation? What can we do with all these players who are sitting around? Sometimes they're not just, there's idle time in the game. And because what this community has done before, they know gamers can be engaged in citizen science. So what the question was, the question of CCP is, what can we do for COVID? Well, flow cytometry is a technology that can have an impact on COVID because we're interested in what happens in the blood when we have an infection. And flow cytometry is, as you all know, the technology to do that. So we got together and we said, let's teach gamers how to do flow cytometry. And so they put up the tutorial together in game. This is a screenshot for that to make them understand the background of the technology, why it's important, and how to gate. Now, these people don't know anything about biology. They don't know what a T cell population is or an NK cell population. And there is no hope of ever being able to tell them the depth of knowledge that the people listening to this today have about the intricacies of why we're gating what we are and what it means and why we're drawing the gates. But the hypothesis we had is the process of gating can be done without understanding the biology behind it. Because we're just trying to find patterns in two-dimensional plots at the deepest level. And our eyes are supremely turned by thousands and thousands and thousands and thousands of years of evolution to find patterns. That's why we're not getting eaten today by saber-toothed tigers, right? We can recognize that this is coming to eat us. And those who didn't, they got called out. Those of us who could find patterns of in flow cytometry data survived, and we're having kids. And that's why that's how evolution works, right? And so these patterns are recognizable, but because they're never going to understand a gating hierarchy, and we can't give them some flow cytometry data and expect them to go through trying to find the populations in the way that we want to find them. It just would never work. But if you give somebody a two-dimensional plot and say, draw circles around the clusters, that's not a hard problem to explain to somebody. And we put together some tutorials to explain, these are the kinds of things that you're trying to optimize. This is what a tail looks like. This is what a peak looks like. And this is how you want to draw the dots. And we kind of gave them a little bit of instructions, and people had to go through that and get trained up on some examples before we let them loose on real data. And as they're going along, we hit them with some gold standards every now and again to keep them honest, so they're just not blindly going away. And the thing is, people care, right? We launched this right at the start of COVID, and it's still going on today. These gamers are like us, right? COVID sucks, and they want to help. And so we had the hope that they're really going to try their best, and they have been. But how to get around this problem of not knowing what to gate everything. So rather than just go through this one plot at a time, gating the 3 or 4 samples at the top level with the specific bivariate plot is that you want to look at first, we have them look at all the possible combinations at the very top level. So if you have a 40 marker panel, we look at all combinations of those 42 at a time and say, hey, gate everything. Because unlike us, where we have limited time, we have infinite number. We're writing Shakespeare here, right? And so if you have an infinite number of monkeys, you can write, eventually they type enough typewriters will get Shakespeare out, right? Same thing here. Because we have so many engaged players, we can gate all possible bivariate combinations of gates and just go through the data all at the time. And then at the end, we can we're not only going to get the populations that you wanted to find as a scientist, because they're going to be in that mix, we're going to find everything. And so this gives us the opportunity to do biomarker discovery with manual gating at an enormous scale. So the way we're doing it is we went through PubMed, went through Flow Repository, Cytobank, reach out to authors who are doing COVID research saying, please give us your data. It still makes me so sad in today's world, even with COVID, how many publications are out there in the literature where people have published flow cytometry data with COVID and have not shared their data. It pisses me off. Stop that. If you see it, call somebody out. There is no reason why people should not be sharing their raw data at all. That's what science is about. Anyway, I write to people saying, please share your data. We get the data. We look at all bivariate combinations of markers. We hand that over to our collaborators at McGill, who format plots in a way that's gamified for the users, make the colors nice so it fits into the architecture of EVE Online. That gets handed over to Massively Multiplayer Online Science, which is a company that's devoted to citizen science. Who knows these things exist? Which then pipes the data EVE Online, who show that to the players. And then they gate everything on that first level, all those first level bivariate plots, hand it back to us. We do some fancy math, and then we're going to iterate through all the levels. Now, some of that fancy math is because we have so many players involved in this. To get the best possible result, we take 50 solutions, 50 different players for each plot, do some fancy math algorithm to get the best combination of those 50 to get something that as closely approximates the truth. So, here on the top, very top, this player didn't do a very good job. These players down here did a better job. And, we're right now working on the algorithm to get the best one we can. Again, based on some gold truth samples we can, to weight the right number of players, to try and guess, working on to get the right number of N by doing some machine learning backwards feed forward stuff. So, if we know how many clusters are in there, based on some training, the players are trying to do a good job. Are we getting pretty close to the truth? It's amazing. We really are. I'm going to just cherry pick some examples here for you. On the left-hand side is the raw data. On the right-hand side is how the gamers gated it, 50 piled up together. It, to me, looks as good as we're going to get by hand. Now, at the end, then we can, after we get the cluster on the right where all the events have been associated, then we can draw a convex hull around that to get the gate on that population. But the important thing is we can enumerate the cells that were done and placed in each gate. Another example, 3 populations here. I think they did a pretty good job. Of course, all the ones I'm going to show here, they did a good job, but these weren't, it's really across the spectrum. We've seen players do a really fantastic job at the gating. These kind of tails. Now, would you actually want to split that out? Now, in the game, we don't actually show them what the markers are, because again, that's not important to draw the gates. And biologically, you may actually want that tail to be combined with that population, because in your biology, that you think they're all the same. There's reasons to believe, I think, that maybe it shouldn't be, because we can expect populations to have a normal distribution, and that looks like something else is going on. Now, in some cases, you might want to join tails to the main population. Sometimes you may not, but unless you can break these components apart, you never have the chance to put that back together. So we've taken the approach that, tails and things we want split off, and then in specific biological context, we can put them back together. But there's only a few component pieces, and people seem to be doing a really good job. Here's another thing. There's this little bleb coming up off of here. Is that part of this population? Maybe, maybe not. We say probably not. We can argue about this over beers later when I see you guys all again. I miss you all. But we kind of train the players to split all these off and have these center clusters, and stuff that comes off to the side, we want those put into separate populations. And we're now out rolling out visualization techniques to show them that, for example, this shoulder is somewhere you might want to cut it off from the main population. So it's working, and so just showing you some numbers. I can't show you all the data, and we'll talk about just in a second about how much data we have. But this is about 1,500 different examples. We know what some of the gates are easier. We know what some of the gates are hard. We spend a lot of time going through lots and lots and lots and lots of player examples to make sure everything's working. I don't know how many thousands and thousands and thousands of things my lab has looked through to make sure that this is all working. Here we're using the adjusted rand index for some math reasons I can talk about later if you want. And with the ARI, there's some accepted scores for what poor, moderate, good, or excellent thresholds are. And so on easier gates, people are doing pretty much excellent compared to gold standards where we know what the answer is. And when I say we know what the answer is, that's where we've used flow density, which hopefully I convince you is a really awesome algorithm to do the gating. And that's our gold standard that we're comparing them against, that they don't know what the results. And then on some harder gates, this is the distribution again. It's not as good. Harder gates are harder to gate. That's why they're called hard. But again, we've seen what some of the challenges people have, and we're rolling out better visualizations now to target them and to give them better instructions so that they're going to do a better job at the end. So we started it. This project launched right at when COVID started. It was a huge effort at CCP to get this up and running as fast as we could. Between March is when it started, and they had this out to the players in June. Since then, up until now, there's been 26,000 people who've done gating for us. On average, it's about 260 classifications per user. One person really likes gating. They've done 44,000 gates for us. So far, up to now, we have 88 million bivariate plots gated. Of those, when we started this project, there wasn't a lot of COVID flow cytometry data in the pipeline. It takes a while for biology to catch up. So we're using other data sets to get going on this. So we're looking at data sets that are related to COVID. For example, people in nursing homes who had been affected by flu, because we thought that might be interesting comparisons at the end to start people going. And then it helps us sort of benchmark. We also wanted to benchmark data. So we're using data where we've had a lot of experience in my lab. We had gold standards, so we can make sure that players are doing a good job before we start and get all the bugs worked out before we start working on the COVID data. And that's worked really well. And now we're really transitioning to pure COVID data. So where we are today, I think we're in a really good spot. When this project is done, our goals are we're going to release everything that we've done again. In the spirit of what everything my lab has done, back to the community. So all the data that has been done by the participants is going to be released back to researchers. All the data that on COVID is going to be released back to the community, to mine. We're going to widely share the raw data. So one of the things like I complained about is people haven't shared the raw data. So I nag people to give me the data, and now we have a way to share that through Flow Repository because they didn't share. It's hard to share data. I get it. Do the work. We're doing the effort for them with COVID to get some of the data out. And now we have so much data. We have the ability, we believe, to do the next generation of machine learning, artificial intelligence algorithms that are going to replace what we're using now, which is supervised, which my lab is using now, and other people who are using our stuff, the supervised analysis with machine learning algorithms, because we're going to have so much fantastic data that we can train algorithms because we're showing the algorithms, this is how people gate. And we know they're doing a good job because they're matching what has been done now. And this is going to change the game. This is going to change how people gate. It's going to be it's going to sound like somebody you might know. It's going to be fantastic, right? We're going to use, because it's being trained to gate independent of biology on the shapes of flow cytometry data across a huge spectrum of different data sets. So this is going to be applicable to everything, and it's not going to have to be tuned for every data set that comes up. So it's going to help us for COVID, for cancer, immunology, vaccine, everywhere flow cytometry is going to be used. We hope that, and because we're going to release this to the community of people who are real experts in machine learning, they're going to be able to have now the data sets that can allow them to develop the best algorithms. It's going to be fantastic, and nobody will ever have to gate again, and that's going to make me very happy. Huge number of people involved in this. The societies that have supported this, companies have given their time of the resources of their people to help contribute. AWS is who's going to help us release all this data back to all the users out there, to all the communities, to do all the experts in machine learning to do their work. I really have to thank my lab for all the effort that they have done. Fantastic group. It's been a lot of time. This has been a full-time job for a lot of people for months and months and months. And this is not going to go away. We expect this project to continue for about another two years. It's great that we're going to have the vaccine, but the research on COVID is not going to stop when we have the vaccine. We want to understand how it works, what's going on. Flow cytometry is going to be so important. So many experiments can be done. So much data is going to be generated, and this is going to feed into our pipeline. Thank you very much.
Thank you, Ryan. This is pretty impressive. So we're going to move very quickly to the panel discussion. We're running a bit late, actually considerably late, and we still want to go for a break to thank our sponsors. So I'm just going to invite the rest of the panel, Deanna, and I think we have lost Tim. Tim had to go to another meeting, so he won't be able to show up. We have time for 2 or 3 questions. your talks were really impressive, and we have a lot of questions. So I'll try to just really go to some of the more important questions that weren't addressed in your talks. For instance, I have here a question about composition and unmixing, because it's often referred to as a visualization tool. Has there been any work toward using uncompensated data in computational analysis? And I'll open it up for both of you.
The first thing that we do is complain when people have an uncompensation, at least for our work. It's bad data in, it just does, it sucks. Don't do it. You have to, and if Mario Roeder was here, he would be throwing things at people. So you have to compensate your data, and you have to do it well, because if you don't, everything falls apart. And we see it, and we see it. We see the impact of that, at least all the analysis we do with everybody we've worked with. It sucks.
Right. So, and are there more automated tools for compensation out there, or do you think we've reached the
if you're willing to spend the money, right? It takes money. You have to have the proper controls. You have to run them. You have to do all the FMOs, and people don't want to do that.
Right. And actually, this brings me to another question about scaling. And Diana, you did refer the importance of scaling. So the question that came up was, when you're trying different scalings, how do you know which is going to be the best scaling for the different tools that you have available? And is it going to depend on the tools, on the algorithm?
Is it what? Say it again.
Is it going to depend on the algorithms that you use?
What I've seen so far is that there is a certain cofactor that is going to work depending on the technology you use, right? Mass cytometry around 5, flow cytometry or conventional one around 150, and around 6,000 for spectral cytometry. And in general, we check the scaling marker by marker, and we might not have to adjust things that much. But depending on how much background you're getting, on the spread in your population, but the rule would be symmetrical distribution of your population around 0. It's also going to be useful to know from the biology of the population. So if you don't expect to have that separation for your CDAs, as I show, then it's a scaling issue. I've seen the benefits of adjusting the scaling marker by marker when there are issues, for example, TCR gamma staining with a lot of background, where the separation for your negative for your positive is going to be highly influenced by the scaling. And then that's going to also influence your ability to identify that subset with low frequency for the TCR gamma delta. So I guess it's going to be case by case. In general, we have a no-magic number that would feed a data set depending on the technology you use. But I definitely think that adjustment, it's important, or has been important in certain data sets.
Yeah.
Can I just chime in on that?
Yes, absolutely.
It's most important to compress your negatives so that they don't have the same separation between the high end of your negative and the low end of your negative as the separation between the negative and the positive. Because when most of these algorithms, they take channel space, which is just the entire distribution across your transform data space, and they bin into maybe 4096 bins, and they use that as the input. And so visual representation and how tight your negatives are is going to make a difference. So increasing the width basis function to a higher negative number is going to compress that data so you don't get the bifurcated winged butterfly around your negative. That's going to be awful. But compressing that negative is most important, whether you use an ArcSine or a BiX transform. ArcSine is nice because you can get those first couple decades to squeeze down into a nice tight negative peak. So those cells are considered similar to each other along that parameter. And you really focus the algorithm on finding the things that are really positive, not things that are on other two sides of your negative spreading distribution.
Exactly. Well, thanks, Tim, actually, for being able to join. I actually have a question for you. So you mentioned TriMath, right? And there's somebody here asking if there are any algorithms that allow you to weigh lineage parameters. And the rest of the question is, in some cell types, the parameter that you use for identifying the population has less expression than activation markers. Or would you suggest running the algorithm with the gated lineage positive?
You could do that, but basically, if you have something that's really low expressed, like a 1 log difference between the negative and the positive, it's going to be weighted less than something that's 3 log positive. So in order to weight that more, you can change the scaling. You can take away that extra white space on the top end of the range, spread out that lower log distribution, and essentially weight that particular marker more. Because again, it's all based on the visual distribution of your transform space for each marker.
Right.
So I don't know of an algorithm that you can automatically set, oh, weight this one over that one, but certainly you can change the range and distribution to weight different markers when they go into the algorithm.
Right, thank you. And Ryan, of course, everyone was very interested in EVE Online. Again, quite impressive, this effort that you guys have been doing. One of the questions was, where do we play this online? So I think
you can play it now. It's free to play. So you can go online. You don't have to pay EVE any money. You go there, look for Project Discovery. You can log in and gate.
Right. And I believe you can play it at work, right? Because it is work.
It is work. Right? Don't play EVE, the game, because the only way to win at EVE is to quit. It's highly addictive. There's people who've been playing that game for 10 years. So don't start that. But the flow geometry stuff is all there. And if you notice anything that looks wonky, let me know, please.
Okay. And so a final question. We're really running so much out of time. I would love to keep on this discussion. But there was a question about normalizing data. So this any of you can answer. Is it possible to normalize data that was collected on instruments with different configurations using the same panel? And also thinking of longitudinal studies, right? How do we normalize data using these more sophisticated automatic algorithms? Anyone want to?
It's possible, but I don't know if that's a real valid comparison. If you're going to do multi-cohort studies that are processed in different locations, you should probably try and standardize the instrumentation as much as possible as a first step. And then batch effects are going to occur based on processing differences on who the technician is and how vigorous they are with their pipetting and stuff like that. And those can be overcome to some degree by something like a cytonorm algorithm, which clusters and then normalizes across the different runs. But you need to think about that when you prep your samples and have that common control that's the same from every single week to week. So you may need to process a single Buffy code, send out samples that are identical to each of the sites, and have them run that control with every run so that you have some standard to compare against. Otherwise, you're just kind of pushing the cells together and potentially losing biological variance and variability that is meaningful to you.
Mm-hmm. Right.
Just quickly, and I think it is important to identify what are the sources of variation through the whole process, starting with sample collection and even the design of the experiment, especially for longitudinal studies. So know what the variations are, what are the potential sources of variation, and starting again from the pre-analytical phase, acquisition, and even data analysis. So if you want to integrate multiple data sets, and if you want to try to at least identify that there are batch effects and then try to find strategies to remove them. So what we know is what causes batch effects. It's differences, for example, in your reagent lots, in the way that, as Tim mentioned, the way the technicians or anyone is preparing the samples. Changes in your instrument. There was a PM in the middle of 2 runs for a particular study. Changes in the staining protocol and how you store the sample, how you transport it, the anticoagulant that was used, time, temperature, all those things, instrument performance. So now that you can define what steps in your workflow are going to be sources of variation, the point is how you can prevent that. So effective planning in your study, it's a critical point. If you are going to do a long study, you need to have secure lots of the same reagents, same antibodies. Same thing if you decide to use a reference control biological reference control to have cells every time you run your samples, enough cells and in the same sample every single time, at least the same donor. Also, make sure you follow the same protocols every time, that you have optimized them well, that you ensure your instrument SQC. And if you want to decide different strategies, one is adding the normal donor or including a normal donor in your batch. Another one could be spiking cells that you have pre-characterized and that can give you a way to normalize. The other one would be barcoding the samples. But I guess the point at the end is having a way to identify if there are problematic batches. How you would do that? Multiple strategies as well. Simple one is just overlaying your histograms for the different samples and seeing if there are variations in the intensity. More sophisticated one, use the FlowSum and UMAR or the tools that Tim described to see if our batch effects. Right? Just cluster your samples together and define that. And even more sophisticated tools, cytonorm, batch adjust, FDA norm, gas norm, that will allow you to create landmarks for every marker and register curves and normalize that signal with the caveats that we know are there, is that you might risk removing your biological signal. The biological changes that are driven by your experimental design and conditions might be removed or blunted by this normalization processes. So just be careful and try to plan for everything from sample collection till data analysis.
Yeah, at the end of the day, these unsupervised methods are not going to be magical, right? We have to really take care of all the standardization before, right? Data analysis. So yeah. I actually have several questions of mine, but especially regarding sorting, but we really have to move on. So I'm going to thank you all. This was an amazing panel. I think it was, from the feedback we got, it was really, really useful. So we are going to take a break now. We're going to take a 10-minute break. And I want to start by thanking, all of the sponsors that really supported FlowTex this year, even though it's a virtual meeting. And, we apologize for not having as many breaks as we predicted, but I think it was worth to have all these discussions and presentations. So it was really a service to science that we're providing here. And again, part of the support came from you guys, from the sponsors. So thank you very much, and we'll see you in 10 minutes. Thank you.
Thank you all. Good luck in your experiments. Take care.
Welcome back from break, everyone. We have gotten to our last talk of the last day of FlowTex 2021, and I would like to introduce Dr. Florian Mair. Florian obtained his Master's of Science from the University of Vienna in Austria in 2008 and his PhD from the University of Zurich in Switzerland in 2014. Currently, he's working as an immunologist at the Fred Hutchinson Cancer Research Center in Seattle, where his main interest is dissecting the phenotype and function of human innate cells in non-lymphoid tissues during steady state and disease. His focus specifically on myeloid cells, and in 2018 published one of the first 30-parameter OMIPs for the phenotyping of human dendritic cells. Over the past decade, Florian has been working extensively with different advanced cytometry platforms, conventional, spectral, and mass cytometry, and recently developed an interest in single-cell sequencing technologies. Since 2017, he's an Isaac Mary-Lou Scholar and has actively engaged in teaching flow cytometry courses, in particular systematic panel design and analysis of high-dimensional cytometry experiments. With that, we'd like to turn it over to Florian.
Thank you very much for the kind introduction and also for having me. I feel really honored that I get a chance to present my data here. And it has been real fun to listen to all the talks this morning. And it also made me realize how much I miss all the in-person discussions that we typically have about these topics at CYTO meetings or other in-person meetings. So The title of my talk is Single-Cell Profiling of Human Inflamed and Tumor Tissues Reveals Therapeutically Targetable Tumor-Unique Signaling Networks. And this is basically a project that has been going on for a while and is kind of utilizing all these kinds of different techniques and analysis approaches that you heard about this morning, but more focused on the biology and what we found. I'll start off with a brief introduction into the biology of immune cells that are resident in human tissues. If you're studying the human immune system, for a long time, people have focused on the blood because it was easily accessible, and it was also thought to kind of represent the state of the immune system. But over the past decade or so, it has become evident that immune cells can take up permanent residence in non-lymphoid tissues, basically in every tissue, and that these immune cells have very distinct functional and phenotypical characteristics compared to their circulating counterparts in the blood. The best studied example of these tissue-resident immune cells is the TRM, the tissue-resident memory T cell, which has been well described by several groups and has basically been found in every non-lymphoid tissue that you can think of. And TRMs have various roles. Most obviously, they are kind of the first line of defense for invading pathogens and kind of mount an early immune response. But in the meantime, it has also been described that these TRM cells have various roles in autoimmune diseases or in other inflammatory disorders, but also in the anti-cancer immune response. Another immune cell that is typically found in peripheral non-lymphoid tissues are myeloid cells. In this particular cartoon here, you have only the dendritic cell represented. That's an immune cell subset that we as a lab are particularly interested in, and I want to take a deeper dive into these dendritic cells and other cells of the same family, which are basically all under the umbrella of myeloid antigen-presenting cells. These comprise macrophages, dendritic cells, and monocytes. And these cell types, they would be present similar to TRMs in basically every non-lymphoid tissue at steady state. And what people would most often think about when they hear about these cells is that those are cells presenting antigen in the sense that they initiate the T cell response. That is, of course, very true. But another very important role of these cells is that they provide cytokines, chemokines, and other co-stimulatory or co-inhibitory signals to the T cells that are present at the local tissue site. And this in turn has critical effects on the function that these T cells will exert at that respective tissue site. So all of this is happening in steady state to maintain immune homeostasis, but obviously if you're having a tissue injury or another insult to the tissue, like for example an invading pathogen, then the function of these myeloid antigen-presenting cells will change, and this will in turn, of course, influence the function of the T cells in that tissue. The reason why we are interested in all of that is actually cancer immunology. And you might wonder why I'm bringing up this aspect of general tissue inflammation. Well, the reason is if you think about a growing cancer or an established cancer, there is, of course, the tumor component to it, but there is always some kind of general inflammatory reaction going on in that tumor. And we were basically wondering whether we can gain a better understanding of the immune response that is happening in solid tumor tissue if we take into account that we should not only think about immunology in terms of healthy reference tissues, but about the changes that could happen in a non-malignant tissue that is undergoing an inflammatory process. That's basically the question that we asked. Can we define human tumor-unique APC and T-cell phenotypes by comparing them not to healthy reference tissues, but to inflamed tissue samples? In a way, that's a hard question to ask because obviously you can't get every sample easily from a human source, but we were lucky in a sense that we were having access to quite a unique cohort of samples. So the way we tried to do that is basically we were collecting oral mucosal tissue samples from people that were undergoing routine dental surgeries for inflammatory conditions, such as, for example, peri-implantitis or other chronic inflammatory conditions of the oral mucosa. Then we were also collecting squamous cell carcinoma tumor tissues from the oral cavity, which falls under the umbrella of head and neck squamous cell carcinoma. So these were solid tumors for which primary treatment is actually surgical resection. And those are obviously not from the same patients, but from the same tissue site. For both of these tissue sources, we collected matched blood for each donor as kind of a reference control for the state of the immune system in the peripheral circulation. Then we pursued basically two parallel analysis approaches. We tried to do like a comprehensive cytometric profiling of all antigen-presenting cell and T cell subsets in these samples using a 30-parameter T cell panel and a 30-parameter myeloid panel, which back then felt great. By now we are kind of a little outdated, but working on it. And we deployed various computational analysis pipelines. The one that I want to present to you today is based on a forest-based analysis approach termed FAUST. In parallel, we did some single-cell transcriptomics, which will be basically the focus of the second part of my talk. And the underlying concept or the ultimate goal of that was to identify tumor-unique crosstalk between APCs and T cells. So I'll start off with the cytometry data up here. As a first thing, we had a look at the T cells because T cells are typically considered kind of the prototypic effector cells that are required for a successful anti-tumor immune response. And if you've been following the literature, there is of course a lot of success about anti-checkpoint immunotherapy, and also about T cells with a tissue-resident profile, because it has been speculated, or it has actually been reported, that cells with a TRM phenotype are kind of predictive of a positive anti-tumor immune response. However, when we started looking into our samples, so throughout my presentation, you would always see the matched peripheral blood shown in blue, the inflamed oral mucosal tissue samples shown in orange, and the tumor samples from the oral cavity shown in red. And when we looked at the expression profile of CD69 and CD103, which are these prototypical TRM markers, we basically saw that both the double positive CD8 TRMs and the CD69 single positive TRMs showed comparable abundance between the oral mucosa and the tumor samples. That was maybe not so surprising, but we were pretty surprised when we started looking at PD-1, which is kind of this prototypic molecule of T cell exhaustion and is also the target of various immunotherapeutic approaches, when we basically saw that the expression level of PD-1 in our tumor samples was basically indistinguishable from the expression level in our inflamed oral mucosal tissue samples. We also looked on a smaller cohort on the expression of the transcription factor TCF1, which has been postulated to be kind of critical along the path of development for exhausted T cells. And again, we saw that the expression pattern between our two tissue sources was kind of comparable. In our T cell panel, we had, of course, a bunch of other markers, and I'll spare you all the raw plots, but kind of summarize that data in a heat map here. So here on the left-hand side, you can see a heat map of average marker positivity within CD8 T cells. And over here, you can see a heat map for the average marker positivity within the CD4 non-Treg population. The upper row represents the blood, and the middle row, the oral mucosa, and the lower row, the tumor. And you, without looking at the actual numbers, you can probably appreciate that based on the expression patterns, the two tissue derived, so the T cells derived from the oral mucosal tissue or from the tumor tissue are overall very distinct from the peripheral blood, but between the two tissue sources are remarkably similar. So we were kind of a little surprised by that observation and concluded at that point, well, there is a relatively large congruence. And for time reasons, I'm not showing you the cytometric analysis that we did for the antigen-presenting cells, but basically we also observed remarkable similarity between these tissue sources for all APCs that we analyzed. So at this point, we concluded the immune infiltrate in inflamed tissues and tumor tissues is surprisingly congruent, and this also includes T cells with an exhaustion phenotype. So the obvious question is then, that I posed in the beginning, which T cell phenotypes are then actually unique in the tumor microenvironment relative to general tissue inflammation? And as you heard during the fantastic data analysis session today, there are, of course, a ton of tools that you can deploy to do that, each of which comes with specific advantages and disadvantages. For the purpose of today's talk, I want to highlight this tool here that we used. It's termed FAUST, which stands for Full Annotation Using Shape-Constrained Trees, and has been developed by collaborators here at the Fred Hutch, led by Evan Green and Raffle Gotardo. And in a sense, it's somewhat similar to other tools that have been described here. So what it does, it would take your sample and it would grow a random forest of gating trees, like basically all different gating trees that you can think about, and it does so by automatically setting boundaries within your samples based on the density distribution of your data. There are some modifications that you can do to it. And what it does in the end, and that's kind of what was interesting to us, you can use a statistical model to basically take like two conditions. In our case, it would be the two tissue conditions where you say which phenotypes that are present in both samples show differential abundance. And that's what we did. And the output are actually very nice histograms, which I'm not showing you, and in a sense, very boring kind of marker combinations. And I won't walk you through all of these, but among the top 10 phenotypes that we discovered, there were two things that were interesting. The only CD8 T cell phenotype that we found as enriched in tumor tissue was actually a CD8 T cell expressing the tissue residency marker CD103, and then various combinations of PD1, CD38, and TIM3, which is also a typically exhaustion marker, while in turn, the CD8 T cells that just plainly expressed PD1 together with various other combinations of tissue residency or memory markers showed decreased abundance. But the top hit that we found as enriched in the tumor was a CD25 positive CD4 T cell phenotype, which is obviously a regulatory T cell. And that by itself is not very surprising, but what we found is that it was different subsets of Tregs that express different combinations of the tissue residency marker CD69, ICOS, HLA-DR, TIM3, and CD38. That was actually the phenotype that was basically had the highest statistical value in our analysis. So we went kind of back and tried to go back to our manual gating, because I still think that's a nice way of kind of checking what's in your data. Up here, you can see that this strong enrichment of the Tregs in the tumor tissue is, of course, statistically significant. It's by no means surprising. For the purpose of our panels, we define Tregs as CD25 positive, CD127 negative cells. Down here, we did some verification. If you look at those CD25 positive, CD127 negative cells, all of these express FOXP3, high levels of CTLA4, also CD39 and TIGIT. So I think it is safe to assume that these are bona fide Tregs that are suppressive. So when we looked at these different marker combinations and tried to kind of boil it down to something that could be more easily reproduced, for example, for sorting or for subsequent functional assays, we noticed that looking at the combination of ICOS and HLA-DR was actually sufficient to identify a unique population. Let me walk you through this rather busy slide. So this upper panel here basically shows you for peripheral blood, the oral mucosa, and the tumor tissue, the expression pattern of ICOS and HLA-DR for the Treg population. This here is for the CD4 positive non-Treg population, and this here is for the CD8 population. And you can basically see, if you look up here in the tumor, that this kind of double positive ICOS HLA-DR Treg population, or this ICOS HLA-DR double positive population, seems to be only present in the Treg population and not so much in the CD4 population, and not at all in the CD8 population. Over here, you can see summary graphs across all our data, summarizing the abundance of ICOS positive, HLA-DR positive cells among the Tregs, and down here among the non-Tregs. And as you can see, this is the only part of the T cell compartment where we can see significant double expression of these two molecules. So at this point, we concluded most of the T cell infiltrate in the tumor is actually phenotypically similar, and the ones that really distinguish the tumor from the non-malignant inflamed tissue microenvironment are these double expressing Tregs. In the beginning, if you remember, I was pointing out in the end, we are interested in how does the crosstalk between APCs and T cells regulate local T cell function. And so we were asking the question, which antigen-presenting cells could potentially be instructing those T cells, and which of these APC subsets are unique to the tumor microenvironment relative to the general inflammatory microenvironment. The cytometry data didn't help us much here, and this is basically where our single-cell RNA-seq data comes in, and I just want to walk you for a moment through the experimental setup that we had here. We had the same tissue sources. We had actually 3 to 4 donors for the inflamed, 3 to 4 donors for the tumor, and in this case 6 to 8 matched blood samples. In order to ensure that we are also capturing kind of rare populations, we enriched the pan T cells and the pan HLA-DR population. And then we ended up with a really nice and large data set. So we have a total, after QC, we have a total of 150,000 cells, which to best of my knowledge is one of the most comprehensive single-cell RNA-seq data sets from human tissues, not peripheral blood, but from actual human tissues. And we analyzed that data with various different approaches. We used Harmony, which is an algorithm for kind of integrating batch effects across different single-cell RNA-seq experiments. And then we did a variety of different exploratory analysis approaches. I'll start off by just showing you an overview of that data set. Over here is kind of an automated annotation of the cells that are present in our sample. We used a package called SingleR, which kind of does this like broad upper level annotation of the cells that are present. And we can find all the different T cell subsets, of course, all the different monocytes, PDCs, conventional DCs, what have you. So this over here would be the myeloid fraction. This over here would be the T cell fraction. And one thing that I want to point out here is if you color the origin of these cells, whether they are from blood, from the oral mucosa, or from the tumor, you can right away see again, similar to what we've seen from our cytometry data, that the two tissue-derived the cells derived from these two tissues, they kind of mingle all together. They mingle separate from the blood, of course, but mostly together. And it's relatively small islands where you start seeing differences between the tumor samples and the oral mucosal, the inflamed oral mucosal tissue samples. That was kind of a good way that this unbiased, truly unbiased approach, if you want to call it that way, kind of confirmed or emphasized what we saw also in our flow cytometry data. So for the next couple of slides, I'll zoom in on this population over here, which represents the myeloid antigen-presenting cells. And if you do that and recluster the data and then look at the heat map representing key transcripts that are known to define these populations, we basically can see all canonical subsets. There are basically 3 clusters that I want to delve a little further into. There were 2 subsets of CDC2s and DC3s, which used to be called kind of inflammatory DCs, which are these 2. They are somewhat related, and we'll talk more about the DC3s in particular. And then there was also this cluster over here that we initially, like 2 years ago, called tissue DCs. By now, there has been a report from the Mair laboratory in New York, published in Nature, last year actually, where they describe these cells as mature DCs enriched in regulatory features. And this is the same nomenclature that we are applying here. So this seems to be a DC subset that is truly only present in tissues and can't be identified in peripheral blood. And it is actually a very, very interesting subset. And they made a very nice description of those. When you look at the relative distribution of these different myeloid populations across our donors, you can see that the frequency, of course, varies, but overall kind of the distribution is relatively similar, with the blood being primarily comprised of like classical and non-classical monocytes and PDCs and CDC2s. And in the tissue samples, you can see that classical monocyte population is reduced, and in turn you have actually a lot of these DC3s coming in, and surprisingly, actually some mast cells in the tumor tissue. Now that we basically mapped out which cells are present, the question that we asked is, okay, how does it look like in terms of functional profile? And for this, we first made this plot here, which is kind of a dot plot where the size of the plot would show you the percent expression of a particular transcript among a given cluster, and the heat map scale would basically show you the average expression level of that transcript. And then we plotted like key co-stimulatory and co-inhibitory receptors like a CD40, 80, PD-1 ligand 2, PD-1 ligand 1, a couple of key chemokines, a couple of key cytokines. And what we noted is that there was actually a strikingly subset-specific expression pattern of these chemokines and cytokines across the different tissue-derived cells, with the MRAC DCs, as reported by the Mair laboratory, expressing the highest levels of CCL17, 22, and EBI3, which was basically not found in all the other subsets. but a bunch of other cytokines and chemokines being restricted to the DC3 and DC2 populations. This is basically the functional pattern of these APCs across the two different tissue sources that we had. But the ultimate question, of course, is what is different in the APCs in the tumor relative to the inflamed tissue microenvironment? And what we did for that is basically just performed the differential gene test across those different clusters. And what we noted then is if we look across these different clusters, for most of these APC populations, there was less than 50 genes that changed in the tumor condition relative to the mucosa condition. And the only two populations that showed significant adaptation to the tumor relative to the general inflammatory microenvironment were the DC3s and the cross-presenting DC1s. That was kind of interesting to us because it implied that those two populations are probably the ones that matter the most. And so we started to look a little bit closer into these differentially expressed transcripts and also the ones that are shared. And this is highlighted here in these violin plots where the upper panel would basically show you the DC3 population and the lower panel would show you the CDC1 population. And this is just a key transcript to confirm that we are really looking kind of at the same cells across the three different tissue sources. Here's kind of CD14 for the DC3s and CLEC9A, which is a key transcript or defining transcript for CDC1s, which is of course found across all three sources. Then there was a bunch of chemokines and cytokines, and I'm not listing all of them here, which basically showed very comparable expression patterns between the tumor samples in red and the mucosal samples in orange, such as, for example, CCL4 and IL-1 beta here, or CXCL8 and TNF down here in the CDC1s. And then there were very few chemokine and cytokine transcripts that actually showed differential abundance. For the DC3s, this was TGF-beta, which was enriched in the tumor, and CXCL16, which is the ligand for the chemokine receptor CXCR6. For the CDC1s, the only two transcripts that we could find that were significantly enriched in the tumor was IL-18 binding protein, which is basically antagonizing IL-18 signaling. And interestingly enough, this has actually been described in yet another Nature paper last year that proposed that this protein can actually be used as kind of another checkpoint regulator of anti-tumor immunity. And then osteopontin, which is, if you do a PubMed search for it, is actually a protein that has been implied as a general negative regulator of T-cell effector function, which also showed increased expression levels in the tumors. So at this point, we knew that there were like few transcripts or few chemokines and cytokines that are indeed enriched in the tumor microenvironment, but we didn't really know how to link that with the T-cell effector function. And then we came across this, this really nice tool that allowed us to address this question: which unique signals are exchanged between APCs and T-cells in the human tumor microenvironment? And the tool that we used is termed NicheNet. Down here is a link to the publication. It's coming from a group that is doing fabulous work at the University of Ghent. So, I mean, most of you will probably know. So Ian is also the senior author on the FlowSum paper. And what NicheNet does, it's really cool in the sense that it takes in single-cell transcriptomics data, and it does not only look at expression patterns of certain ligands and receptor pairs that are known to interact, but basically you can define, so to say, a sender cluster and a receiver cell cluster. Then you can also run a differential gene expression test, which in our case we ran between the tumor and mucosal cell types. And then NicheNet would actually look at the expression pattern of downstream target gene networks in order to infer the activity of a ligand target. Let me give you a specific example. Let's, for example, say you have IL-6 and IL-6 receptor, and both of which are expressed in your single-cell RNA-seq dataset. NicheNet would only infer that that's an active pair if in the target cell population also the downstream known target genes of the IL-6 receptor signaling chain are active. Let's say, for example, certain stats or certain effector genes that we know from previous experiments are induced by IL-6. So that's basically what NicheNet does. And then, as I will show you in the next couple of slides, we ran, of course, some validation experiments to confirm that. Over here is a circus plot that we use to kind of evaluate the data, and that might look like really complicated and overwhelming. So let me walk you through this. Up here, you can basically see the receptors that were found to be expressed on the receiving cell population, which in this case would be the CD4 non-Treg cluster. And down here, you can see the ligands expressed by the APCs. And I basically grouped them into three different groups. Over here, blue is cytokine co-receptors, such as these and these. The green stuff, to be very transparent, this is basically where I didn't really know what to make out of it. That's basically why I labeled it a different color. And then the ones that I want to focus on are these orangish ones, which I termed unique and interesting. Why unique and interesting? Well, we found that if you look at the different T cells as separate receiver populations, you will actually see that there are a lot of pathways that are shared, and then there are some which were predicted at least to be unique to the CD4 non-Treg population, to the CD8 T cell population, and most strikingly to the CD4 Treg population. According to NicheNet, the CD4 Tregs in the tumor microenvironment uniquely receive signals from ICOS ligand, IL-15, IL-18, and most surprisingly, IL-1 beta, which is traditionally considered a pro-inflammatory cytokine. Well, at this point in time, all of this is, of course, only inference, but we knew already from our cytometry data that ICOS is highly expressed on those Tregs in the tumor microenvironment. In all likelihood, that's a valid prediction. And for these other three cytokines, we went on and performed validation experiments. This is what you can see here. Basically, we performed an intracellular cytokine staining for IL-1 alpha and IL-1 beta for the different APC subsets. IL-1 alpha and beta are not expressed at all on the PDCs, but they are expressed at high to intermediate levels in the conventional DC population and in the monocyte macrophage population in the tumor. We also performed Luminex analysis, just like on direct flash frozen tumor tissues, where you can see that there is quite a large spread, but that there is definitely IL-1 alpha, IL-1 beta, and also IL-18 present in the tumor microenvironment. So at this point, we knew that the ligands that were predicted by NicheNet seemed to be indeed present in the tumor microenvironment. And the next question that we asked was whether the corresponding receptors are there as predicted by NicheNet. And since we were most intrigued by this IL-1 alpha, beta, IL-1 receptor signaling axis, we started looking for that. Luckily, there is actually an antibody available for the IL-1 receptor type 1 and IL-1 receptor type 2. This one is thought to be like a decoy receptor, and this is thought to be like the active signaling domain of the IL-1 receptor. And when we looked at the expression of these receptors across the different T cell populations in the tumor, which you can see here, those are the tumor CD8s, the tumor conventional CD4s, and the tumor Tregs, and here just like all T cells from the blood, you can see that indeed expression of the IL-1 receptor type 1 seems to be exclusively confined to tumor infiltrating Tregs and not to be found on the other T cell populations. Over here, you can see quantification across multiple donors for the expression of the IL-1 receptor across Tregs, and it's not found in all the other T cell subsets. If you have a closer look at the IL-1 receptor positive versus IL-1 receptor negative Tregs from the tumor, which is what you can see down here in panel F, you can basically see that the IL-1 receptor positive Tregs are also the ones that express the highest level of ICOS and HLA-DR, which is what Faust predicted in our cytometry data. And those Tregs also seem to express very high levels of the IL-18 receptor, and also at least 50% of them express the ligand for CXCL16, which is the chemokine receptor CXCR6. So at this point, we felt that we were able to confirm the predictions that NicheNet found in our single-cell RNA-seq data, and that is that IL-1 alpha and beta is indeed present in the tumor microenvironment, and surprisingly, the expression of the corresponding receptor seems to be specific to regulatory T cells. Well, that obviously brings up two follow-up questions. The first one being, are IL-1 receptor positive Tregs truly a distinct subset of tumor infiltrating Tregs? At this point, I want to point out a more practical problem that we had at this time. I mean, everyone keeps talking about single-cell RNA-seq data, but it's actually quite laborious to make that or to generate that data, and a significant effort and that goes into these pipelines. And you also need to have the samples. So we had two problems here. First, we knew from our single-cell RNA-seq data that the IL-1 receptor transcript detection appeared relatively weak, which is actually very common for many genes. Kind of the dropout rate in typical whole transcriptome or in general single-cell RNA-seq protocols is relatively high. In this case, we knew that we were at best seeing less than tenfold of the transcript than we knew from the surface protein. And then the second problem is, in general, those human tumor samples, it's hard to get them, and they are usually very limited in size, and we, in general, have few of them. And then there was COVID-19 on top of that. So we basically, the technicians that usually go to the OR to collect these tissues from the surgeons, they couldn't go in. So we were kind of cut off from our supply, and we wanted to wrap up this project. So what we did then is, basically what we wanted to get is we needed to have the phenotypic data, and we needed to be able to sort in parallel without losing any sample. And in this case, we were actually lucky because the Hutch just got the S6 sorter. And so we basically, I didn't have much choice. So I basically just took the established 28 color panels that we had and whacked it onto the S6, hoping that it would work and I could sort the populations. So I literally just took my target values from the FACS Symphony, ported it to the S6 just after it has been installed, and just hoped that it would work. And luckily it did. So I'm just showing you here an example for those of you who are maybe more technically interested in how things looked like. So this is basically my reference panel that I used all the time, where I knew how things look like and how well they are working. Like we can see the PD-1 expression, TIM3 expression, and here the Treg population. We can see 4-1BB, we can see ICOS, what have you. And down here is basically just trying to get that onto the sorter. We basically can see the same populations and kind of the same resolution. Of course, things look a little different because of the visualization, but I was actually happy that that worked. And we were able to sort our Tregs and split them into the IL-1 receptor positive and negative Tregs, and in parallel get the phenotypic data acquired for those samples. So what we did basically is we performed, in this case, a targeted transcriptomic single-cell RNA-seq experiment. And I'm not going to show you all the data, but what I'm focusing on here is basically we had 3 donors, and this is compiled data of 3 donors. You can run whatever is your favorite clustering algorithm. That's kind of the phenograph implementation of Seurat. The point being, though, here, if you focus on these violent plots, you can see the blood Tregs in green, the IL-1 receptor negative Tregs from the tumor, and the IL-1 receptor positive Tregs from the tumor in orange here. You can see that, of course, all these 3 subsets express FOXP3. They also express CTLA4. They also express the IL-2 receptor, and they also express varying levels of ICOS. But then there's a bunch of transcripts which seems to be uniquely expressed in the IL-1 receptor positive Tregs in the tumor, depicted here in orange, including of course, IL-1 receptor type 2, IL-18 receptor, which we knew from flow already, CXCR6, CCR8, this is CD39, and ID3 is a transcription factor that has been implicated in the development of a tissue regulatory Treg program. At this point, we felt, okay, these IL-1 receptor positive Tregs are indeed a unique population in the tumor, but the ultimate question, of course, remains, so what, do they show a distinct functional response? Do they do anything different or anything that is unique to the tumor? And it's kind of hard in a sense, because for conventional effector T cells, if someone asks what is kind of the functional response, it's in a sense easy to assess that you can do an intracellular cytokine staining. You're known that they are producing like a bunch of cytokines. But for Tregs, it's a little harder because you either need to do a suppression assay, which is not really feasible with like the limited amount that you get from a human tumor sample. And in terms of effector program, it's not so well defined for Tregs what they do. So we kind of had to go again on a fishing expedition, and this is where we did like another single-cell transcriptomic experiment, in this case combined with surface profiling of proteins, AbSeq or CytSeq or however you want to call it. And we did that actually with stimulated T cells, where we deployed kind of a short-term stimulation assay. So we basically sorted the T cells out of the tumor, and then we did like either a 2-hour unstimulated condition, 2-hour stimulation with cytokines, or 2-hour stimulation with PMA ionomycin, which is basically what I'm going to show you. And then you can take that combined data set, which in this case is coming from two donors, and you start actually see, so this is only CD4 T cells from the tumor from 2 donors after stimulation, and you can start seeing this remarkable heterogeneity in terms of their functional responses. And I know some people don't like heat maps, but I really love heat maps because there's like so much information in there. So this is basically coded up here in unstimulated and stimulated for the different clusters. So this would be the unstimulated CD4 cluster, the unstimulated Treg cluster, and the stimulated Treg cluster. and over here, different clusters from the CD4 compartment after stimulation. And you can really start seeing these distinct functional responses. For example, this here seems to be the cytotoxic CD4 cluster that has been described by others. Here are two distinct CD4 clusters. And you also see that the stimulated Tregs seem to cluster separately from the unstimulated Tregs. And that's where we dig in a little deeper. The nice thing about that assay is that all of you that do single-cell RNA-seq know that sometimes it's really hard to resolve T-cell populations based just on their transcript. And so here we had the protein, and this is surface protein expression after sequencing. So here you can see CD25, CD127. You can see that we really see very nice separation of those two, and we can clearly, just based on protein, confirm the identity of those cells. And down here, you can actually see what we saw after stimulation, and that's kind of really interesting. So this over here, this gray column refers to, in general, unstimulated cells. This blue column refers to stimulated CD4 T cells, and this here refers to stimulated Tregs. And you can actually see that IL-2 and interferon gamma is like, of course, coming up in the CD4s at very high levels, not really in the Tregs. And down here, you can actually see something that was already interesting, kind of CD40 ligand and CTLA4 is showing very differential expression patterns on the CD4s versus the Tregs, with the Tregs expressing very high levels of CTLA4 compared to unstimulated. And also this population that is co-expressing CD40 ligand, which could be indicative of that those are the Tregs that are really interacting with the APCs via CD40. And the last data point that I want to show you here, because that's the question that we wanted to ask, we could actually use, again, the protein data, since we had the protein data in that sequencing or in this multi-omic data set, and can look at IL-1 receptor protein versus ICOS protein and take the double positives versus the double negatives. And what we can see is that the double positive cells by protein, after stimulation, they indeed seem to be expressing higher levels of CTLA4 and 4-1BB up here, and also higher levels of the IL-2 receptor alpha and IL-2 receptor beta, indicative of their activated state. So we do really think that these cells show a differential functional response, and this is like very recent data, so I basically got that just a couple of weeks ago, so we are still analyzing and digging deeper here to kind of delineate what does this tell us about the functional profile of Tregs in the tumor. And I think this brings me to my summary, and I think we are kind of on time. So what I hopefully could show you today is that the immune infiltrate in inflamed non-malignant human tissues and human tumor tissues is largely congruent, including T cells with an exhaustion phenotype. Yet we can use computational analysis approaches. In our case, we utilized FAUST to reveal a tumor-unique regulatory T cell phenotype, which co-expresses HLA-DR and ICOS. Our single-cell data set showed us that specific APC subsets express distinct cytokine and chemokine modules in human tumors, and trying to link that to T-cell function using NicheNet showed us a tumor-unique Treg population that co-expresses HLA-DR, ICOS, and the IL-1 type 1 receptor. And we are currently trying to figure out what this tells us about IL-1 function in the tumor. Of course, this project involved a lot of people. I want to thank AI for the Intersect Fellowship, Isaac for the Mary Lou Scholarship that supported me during the past years, and all the people involved in that project. Most importantly, my PI, Martin here, my coworker, Jamie Erickson, with whom I did basically all the 10x experiments together and some of the Rhapsody targeted transcriptomics experiments. Our co-workers here at the Hutch and the University of Washington, and Aaron from BD for always helpful discussions and support. Over here is my email address if you want to follow up, and I'd be happy to answer any questions that are coming up. And I thank you very much for listening to me, and the FlowTex has been really amazing. Thank you very much for having me, and I hope to see you again in person at next year's CITO or FlowTex. Thank you.
Thank you so much, Florian. It's such an interesting talk and such interesting work that you do. You've been at the top of our FlowTex committee speaker wish list for a few years.
Thank you.
So we're so glad that you were able to join us this year, and hopefully we will be able to see you in Texas at some time. We do have a good amount of questions that have come in, so I want to start with the first question, and it's, can Faust be used to identify other rare populations, or was it specific for this application in identifying Tregs?
No, it can definitely identify other rare population. The issue that I have with it as an analytical approach is actually that sometimes, depending on the cutoff that you set, the populations get so small that you wonder whether they have a true biological relevance. But it can basically, I think, go to as small populations as you wish, as long, I feel it works well as long as you're having like good bimodal separations. But there are a lot of tweaking parameters, which is actually beyond what I exactly know about it. For this, you would need to reach out to Evan Green or read the preprint. I think the accepted manuscript is not out yet.
So what types of quality controls can you use to identify these populations?
I assume that is referring to the cytometry data, if I got it right. Mm-hmm. So basically the way we have our cytometry experiment set up is we have our tumor and other tissue samples, which are experimental samples. And then similar to what has been briefly mentioned in the previous talks, we basically have a reference control, which is basically one Buffy code from one donor, of which we have like 100 vials. And for every time I do a staining, I thaw one of these like reference vials and kind of look at the staining pattern, of course. And if anything is off there, I know that I messed up the staining or something else went south. So we would be able to exclude that data point. And for the actual tumor samples in terms of QC, I try to basically follow all these good practices that have been outlined today by Rui, in that I do actually have not all FMOs, but I have like 2 FM2s that I consider critical that I typically run on the actual samples to kind of, particularly in my case, for example, for the IL-1 receptor, because I wanted to be sure that what I'm seeing there is not like an artifact of spread. Yeah, that's basically the controls that I run, and I feel relatively confident about them. But as has been discussed, there is no perfect control, so you need to kind of use a little bit of your biological knowledge. And I like to use just density distributions and reference populations.
Mm-hmm. So having identified cell populations that are distinct to tumor and not just present in all inflamed environments, what sort of assays would you consider to determine functional differences in the Tregs?
Yeah, I mean, that's the part that is hard in a way because you're having so limited material available. I kind of feel that at this point in time, the single cell RNA-seq or whatever high dimensional single cell assay you can do is probably the best exploratory approach that you can do. In terms of function, if you were to just do ex vivo, I think that's limited. And this is why we started to kind of devising these assays where you take the cells out and you stimulate them. I showed you only the PMA onomycin, but you could, of course, also stimulate them with different cytokines, which is what we are doing now, and then perform these transcriptomic assays and see what changes in their expression profile. And at this point in time, at least, I feel that's the only assay that is feasible to determine their function, because everything where you need to do bulk, you just don't have enough material and the readouts would not be sensitive enough. I know there is this cool company somewhere in California, I think it's called Berkeley Lights or what have you, that actually allow you to do these kind of assays where you get the readout from single cells after stimulating them with cytokines. So it's only hearsay that I know about that, but it sounds like amazing. Maybe that's where we will go in the future. But right now what I'm doing is basically single cell transcriptomics after stimulation. And I hope that this will provide functional insight.
Berkeley Lights, I believe, is one of our vendors. So that's good to hear that you haven't what they're doing. Have you done this kind of comparison in different tissues or tumor types? Have you looked at other tissues that have more or less tissue resident immune subsets?
So we haven't done other inflamed tissues because, as you can imagine, that's actually really hard to get your hands on. I think the best tissue source that exists is this kind of deceased organ donor source from Dana-Farber, where you might be able to call some tissues inflamed, but we haven't been able to get access to that. We've done other tumors though. So basically we can see the very same cells in breast cancer tissues. That's the only other tissue source that I've analyzed at this point in time. Unfortunately, we weren't able to get access to any others. If you know how to, shoot me an email.
Yeah, absolutely. Do you think you can integrate Nietzsche with protein data using proteogenomic approaches like CytSeq?
Can you just repeat that? I think I didn't.
Do you think you can integrate NicheNet with protein data using proteogenomic approaches like CytSeq?
I mean, I'm sure you can. The way the package is written right now, it wouldn't really. And I think it wouldn't help so much as long as you're relying on antibodies, because all kind of, or many of the ligands that go into that analysis are, you can't really stain for them in a CytSeq approach, because it's like, I don't know, CXCL16 or IL-1, which for all of which you would need to do intracellular cytokine staining, which is not compatible with the CytSeq AppSeq assay. I think they just released a way how you can do basically transcription factors. But I think that would not work. However, there are these, you can basically use the CytSeq data to map it back onto the single-cell RNA-seq data for improved identification of canonical lineages by surface markers. And I think that's probably what should be done in the future. And yeah, so I think you can to a limited extent.
Very nice. How will this type of multi-omic data impact treatment protocols? And are you looking at drug targets?
Drug targets, I mean, well, yes, ultimately we hope that we are able to identify druggable targets, and whether the cytokine receptor that we identified is druggable or not, I think depends on like a lot of additional experiments. At this point in time, I wouldn't be able to tell you, but I hope that we would be able to perform follow-up experiments to address that.
Have you thought about following existing clinical trials using this approach?
Following in a sense like using samples from clinical trials, I assume that's the question. We've thought about that. I think it's beyond my bandwidth of what I can do right now. I think for clinical trials, any kind of single-cell transcriptomic approaches could be very useful. But at this point in time, it's not only a matter of cost. I mean, these assays are expensive, but I really feel it's also a matter of throughput, because the platforms that are available right now, I mean, take, for example, the BD Rhapsody or take the 10x Chromium. Basically, in one run, you're limited to like 20 or 80,000 cells. So you can, of course, take 2 instruments or 3 instruments and kind of beef it up that way. But for a true clinical trial or something like that, you probably, I mean, think about it, even if you would only take 10,000 cells per donor, you do want to use at least like 30 donors to get kind of a response across the spectrum. And then you really end up at cell numbers that can't be processed, like physically processed at this point in time, not to speak about the sequencing cost. And I think what we need here is really a call to manufacturers to have like platforms that allow to process more cells in one run. And then ultimately the sequencing costs need to further decrease, which eventually they will, we know, but that might just take like a couple of years from now.
Yeah, for sure. For the more technically oriented audience, how is the experience in sorting based on the 28 markers, from the live compensation to the gating?
Well, that is an excellent question, and that's what I've been talking about. It somewhat circles back to what Rui was talking about this morning. You really need to have like perfect controls. First thing, I mean, that to be clear. But then on top of that, I think you really need to know your panel and how it should perform, because whatever you do wrong on the sorter, it'll be like, I mean, you're kind of done. I mean, you sort and that's it. There's no fixing afterwards, or fixing in a sense, like no tweaking afterwards.
Right.
So I feel, I mean, I would say it's like perfectly feasible and perfectly fine to do that, that 28 color sorting if you wish to. You just need to have a really solid panel and a solid set of controls and reference samples. And then you can do it. And I think it also depends on your downstream application. If you do, for example, the good thing about single-cell transcriptomics is it's brutal in telling you whether you did a good sort or not. Whatever B cell ended up in your sample, the downstream single-cell transcriptomics will show you, okay, that's a B cell, and then you can still just exclude it, so no harm being done. But if you're doing kind of these assays where you're sorting for bulk RNA-seq or I assume maybe also proteomics, you really want to make sure that you're sorting the right thing. And I think then it really boils down to the perfect controls, the perfect experimental setup, the ability to reanalyze some of your samples, not like maybe the experimental samples, but maybe you can sort another population and make sure that this one is pure and so on and so forth. And I think all of that has to be utilized for a successful experiment. And I think it's true for any high dimensional assay, and just for sorting, also for spectral sorters to come. I mean, you really just have to nail it on the spot. And for this, you need to have the right controls.
Yeah, that'll be really interesting with the spectral sorters coming out. And then we have a final question that came in through YouTube. And they're asking if you've done immunostaining on tumor tissue versus mucosa with correlation to flow data.
We've done, or actually not I have done that, but I had a coworker in the lab that was initially just focused on characterizing the oral mucosa. And she did that. And for the most part, we can see good correlation between flow data and immunohistochemistry, which suggests that the cells that we are isolating are capturing whatever's in the mucosa. But those immunohistochemistry experiments were, of course, very limited in terms of markers. So I can definitely imagine that there are some, let's say, some myeloid cell subsets that are not like captured very well during the single cell isolation, or are just not released very well, so that we are not capturing these. This is probably why we need spatial transcriptomics to kind of really, I mean, I think that's the holy grail to kind of get that resolution inside in the tissue, which I think we are on the right path, but it's not at the same resolution as the kind of the in suspension single cell assays. But it's an exciting time.
Yes. Well, thank you so much for speaking for us today. And like I said, we're hoping that we will see you in Texas sometime soon.
Thank you very much for having me. Have a good day, everyone. Thank you.
Now we will have Joel come on and wrap our final day up.
All right. Well, thanks for everyone attending this year. We really appreciate it. We got a little. It's been a great session. Registration and turnout was awesome. We had 480 registrants this year. We had 1,657 registrants for our science webinar punch out. It's been a great year. I'd like to thank all the presenters, both our YouTube presenters and our science session presenters, that made this happen. Without all your hard work, this wouldn't have been such a great conference. I'd also like to give thanks to all the organizers. We have a great FlowTex team that really helped put all this together, and you guys are all awesome. So finally, I'd like to also say a couple things, actually. One, we are going to do the raffle, but first I'd like to say mark your calendars for next year's FlowTex 2022. Hopefully this is going to be in person. Everything looks like it so far, and that's going to be on February 2nd and 3rd of 2022. So write that down and with a Sharpie. All right.